forward to interesting deliberations such as event, especially being the Decade of the Ocean, gains utmost significance as marine environment serves a variety of bioresources for harnessing natural products. Since the beginning of mankind, nature has contributed to drug discovery for human beings by providing remedial treatments, and one of nature's treasure is the marine biotope. This eye connect on marine derived products having potential industrial applications aims to enable industry and researchers to come together on a common platform and discuss possibilities and issues of collaborations on the relevance of marine organisms as a source of bioactive molecules for biotechnological and pharmaceutical potential, leading to fostering a strong Atmanirbhar Bharat. I now invite Director CSR NIO, Professor Sunil Kumar Singh, to deliver the welcome address. Over to you, sir.
Thank you very much, Judith. Good morning, everybody. Professor Tapas Kunduji, professor at JNCASR and keynote speaker of today's program. Dr. VM Tiwari, theme director of E3OW and director CSR NGRI. Dr. Hemant Kulkarni ji from CSR headquarters. Dr. Sunil Kumar ji, head BDG CSR NIO. Dr. Narsingh Thakur ji, organizer of this program. Dr. Judith Gonjalves, distinguished plenary speakers from the industries, panel members from government organization and industries, participants from industries, academic institutions, National Institute of Oceanography, and NGOs. It gives me immense pleasure to welcome you all in this iConnect event, marine-derived products having potential industrial applications. CSR National Institute of Oceanography. Hello. CSR National Institute of Oceanography is working on various fields of oceanography, and one of them is to derive products, natural products from the living resources from the ocean, and in line with the Government of India policies to leverage scientific and technological research in solving the issues of common man in various sectors. It is essential to have an interaction with industries and entrepreneurs at various levels. In view of this, today CSR NIO is organizing an iConnect event, marine-derived products having potential industrial applications. I am sure this Industry Connect meet will provide a platform for one-to-one interaction between scientists, technologists, industry participants, and funding agencies. As I told, CSR National Institute of Oceanography is a multidisciplinary institute involved in all aspects of oceanographic research. The marine biomes are a rich reservoir of unique life system, making them a promising target for bioprospecting for products and processes having industrial applications. Various reservoirs, ecosystem uses such as deep sea, hydrothermal regions, cold ships, coral reefs, mangrove forest, etc. are hotspots of biology, which can provide various kind of natural products for application in pharma industry, drug industry, food industry, cosmetic industry, etc. The researchers at NIO are working in various segments of marine biotechnology and have an ongoing multi-institutional R&D program, bioprospecting, which is aimed at developing marine-derived products and processes in collaboration with industrial partners. Through this We would like to strengthen our collaboration with our research institutes and various industries for exploring and realizing the potential of marine living resources for societal and industrial application. So I wish a very successful iConnect event on this marine derived products having potential industrial application. Thank you very much and welcome you all again.
Thank you, sir, for your warm welcome. I now request Team Director of Ecology, Environment, Earth and Ocean Science and Water, and Director CSIR, National Geophysical Research Institute, Hyderabad, Dr. V.M. Tiwari, to kindly give the opening remarks. Over to you, sir.
Thank you very much. Very good morning, Professor Tapas Kundu, a very distinguished scientist in the country in the biological sciences, and our today's leader of bringing a connect between the fundamental science and its industrial applications. Professor Sunil Kumar Singh, Director, CSIR NIU, colleagues from CSIR NIU, from CSIR headquarters, panelists, and others who have joined through online media and in physical mode. You are aware that the purpose of this program of iConnect is very large, very vast, and it's not easy to achieve by one individual institute or an organization. And therefore, it is thought that how our science and technology endeavor, the products made by CSIR are the scientists of the Ministry of Science and Technology and Ministry of Earth Sciences can be beneficial to the industry, and vice versa, how industry can be helpful in upscaling these technological interventions adopted, developed by the scientists working in these two ministries. Today, when we discuss about marine living resources, we very much understand that the living resources of ocean is going to be the future for socio-economic sustenance of mankind, and they also happen to be sustainable considering the fact that they can be replenished over a period of time, and with our planned activities of harnessing these resources can benefit the humankind in general and our country in particular. You may be aware, as the director of CSIR NIU has already mentioned, that the institute like CSIR NIU, in collaboration with other CSIR laboratories, and one of them happens to be CDRI, which was led by today's distinguished speaker, Professor Tapas Kundu, had worked in the many areas, including therapeutic molecules, enzymes, natural pigments, and particularly unnatural pigments. Some of the products are already licensed from the work carried out in CSIR, and also biomaterial which we are getting from the ocean. Therefore, bringing industry and one platform is very thoughtful and very planned activity of CSR NIU, and I compliment the organizers and also the scientists, those who are involved in this area, which will be driver for economic progress of the country. I'm very pleased to be part of this discussion, and I'm sure that the outcome, the discussion way forward proposed by the panelists and the researchers, we will be able to take the CSAR mission for making India up. Thank you very much, and I wish a grand success of the discussion, and I'm sure that we will be very much benefited from the presence of the distinguished speakers and panelists. Jai Hind.
Thank you, sir. Thank you for your views and highlighting the importance of marine resources and for your good wishes. We will now have the keynote address by Professor Tapas Kumar Kundu, Professor Jawaharlal Nehru Center for Advanced Scientific Research, Bangalore. I request Director CSIR NIO, Professor Sunil Kumar Singh, to introduce our distinguished keynote speaker. Over to you, sir.
Thank you, Judith. I'm very delighted to introduce our good friend, Professor Tapas Kumar Kundu ji. Professor Tapas Kumar Kundu completed his master degree in biochemistry from the University of Agriculture Sciences, Bangalore in 1989, winning a gold medal for standing first in master degree examination. He was awarded his PhD from the Indian Institute of Science, Bangalore in the year 1995 with Best Thesis Award. Following his PhD, he had a short stint as a visiting foreign research associate in the National Institute of Genetics, Mishima, Japan, followed by a postdoctoral fellowship at the Rockefeller University, USA, during 96 to 99. He joined Jawaharlal Nehru Center as professor in 1999. Professor Kundu has made significant contribution in the area of regulation of gene expression and its link to diseases and therapeutics. He is not only elucidating the mechanics of transcription regulation through the epigenetic modifications, but also targeting them to design new generation diagnostics as well as therapeutics. Over the years, he has published several research papers in many international journals and national journals. Several patent applications from the laboratory have been granted and some are under process, which includes several academically important research reagents with the potential commercial values, some of which have already been commercialized by renowned companies. He is the fellow of all four national academies of India and served as an editorial board member of the Journal of Biological Chemistry from 2011 to 2016. Presently, he is serving as an associate editor of Journal of Biochemistry, which is published from the Oxford University Press. As a director of CSCR CDRI Lucknow, Professor Kundu initiated many relevant academic and drug development programs. Under his leadership, several drug molecules reached to the phase 1 clinical trials and could be licensed to the industries for further development. He also led and completed a phase 3 clinical trials for anti-COVID-19 therapy, which is awaiting for DGCI approval. He was instrumental to establish the Asian Forum for Chromatin and Chromosome Biology and Chemical Biology Society, India. Besides fundamental research, Professor Kundu is also involved in teaching and organizing science outreach programs. His popular lecture on gene diseases and therapeutics have benefited high school and college students greatly. He is a recipient of several awards, noteworthy among which are the Santi Swarup Bhatnagar Prize from CSIR, India Reliance Industries Platinum Jubilee Award, the Sir J.C. Bose National Fellowship from DST, the G.D. Birla Award for Scientific Research, the Ryan Baxi Research Award in the field of medical sciences, basic research, and India Innovation Award given by Mark Millipore, and Sri Om Prakash Bhasin Award in the field of health and medical sciences by Sri Om Prakash Bhasin Foundation, New Delhi. Actually, list is so long and I've already taken a long time, so I'm stopping here. And now I request Professor Kundu to deliver his talk.
Professor Sunil Singh and Professor V.M. Tiwari, my colleagues and very good friend. It's a great opportunity, and thank you so much for almost giving my full CV. And in fact, I remember one word of Professor Mashelkar at this moment. Once you are in CSIR, you are in CSIR forever. So I still belong to the CSIR family, and I'm delighted to see that my family from NIO is here, and my colleagues from the headquarters are also here. In fact, this meeting, iConnect meeting, with the topic of the marine therapeutics and using the marine system for the food and nutraceuticals is a very, very timely. Very timely in terms of the present phase of the development of our country. And also, another reason is that just now we have submitted a big project called the Center for Marine Therapeutics, and where the CDRI, Nehru Center, and different NIPERs, and of course our leader in the field, NIO, is involved. And I am hoping that both the DOP, DST, and DBT will be supporting this project. It is in an advanced stage. And this project has taught us a lot. We had several meetings, and now today's talk, what I will try to do, I will not talk about the project because of the project is yet to be sanctioned, although we have started some work on that. In order to set the gear of the whole meeting, I will just to give an overview of 25 minutes or so, how important is that for India now to really look into our vast sea around us. And also, it is not only the discovery, also to make an affordable therapeutics and affordable, affordable the nutraceuticals from there. Give me 2 minutes, I'll just put my slides. Here, these slides. So I put my screen off. My video I put on. Share the video. Yeah. Then slides. My slides are visible. Hello?
Yes, yeah.
Slides are visible, right?
Yes, it's visible.
Okay, okay. So, I mean, you can ask me the questions in between, but I will prefer that you ask me at the end. That will be better. I have made it little in a popular way. excluding our own work as well as excluding the work of CDRI and other colleagues. So this is needless to say this audience that how important is the marine sources. Now, marine life basically represents the 80% of world biota. The oceanic environment is an abundant source of many things, nutraceuticals, potential candidates with therapeutic functions for our today's talk and discussion. Marine natural products presents a novel and diverse chemotypes. So here, one thing is that it is not only designing the molecule, but some of the molecules, we cannot even think. Human brain cannot think, but the nature has already made it. Even simply understanding the scaffold present in the marine system is, it's more than enough to design many therapeutics. And that is the chemical library comes from the marine sources. And that is amazing. So, what all things can be, it could be therapeutic molecules like anti-diabetic, anti-analgesic, neuroprotective, anti-cancer, antiviral, anti-inflammatory, antimicrobial, and immunomodulators. There are many molecules are coming from there. But here, one point I would like to mention is that that the diverse organisms are there in the marine sources. These are more than some of them are many years, like 1 million to 700 million years ago. A tremendous biodiversity you will find there. You will find there fungi, bacteria, microalgae. Then you will find sponges, mollusks, corals, fishes, many of them. And why these marine organisms are so colorful, why they are making so many compounds, why this is not making for our own use, but they are making for themselves. And largely two different aspects we must keep it in mind. It is for the marine communications and also their defensive mechanisms. Okay, this is the reason, and underwater they need to make it more and more colorful. So just look at these diversities, like starting from sponge, tunicates, algae, crustaceans, fishes, bacteria. Even the bacteria also, they have many types of colorful bacteria, and their genome organization, their defense mechanism, their overall nutrition is very different. So I start with one of the organisms called tunicates. So these are, this gives the many marine products to us, and there are many possibilities to, to explore them. Certain fishes are also release toxins such as bisturmide, and these toxins are also very important. As such, not as a toxin, maybe when I come to the marine, marine natural products inspired synthesis of the drug, you will see these toxins gives us many good painkillers. Okay, and this also, tells us a beautiful biology which is called the symbiotic biology. Symbiotic biology is a very important, in terms of the understanding the microbiota. Okay, and the metabolites which originate from tunicates are also very important. Just to give you the examples, see the range of molecules starting from antiprotozoals to antiretroviral to anti-cancers, many of them we can get from the tunicates. And this I have listed few, and some of them are very importantly, very interestingly being exploited. Okay, valinesin, kunamycin, hanosidin. So these are the compounds, and these are I have also listed here. The there are different activities. Here, one important thing I would like to point out here, that as we are progressing, the drug resistance is, is inevitable, because the kind of antibiotic being used by us directly and we consume indirectly from the different animal products and food products. Because of that, the resistance are developing, and newer antibiotics are very, very important for us to generate. And that cannot be done by just by synthesis and understanding biology. It needs the marine sources. And here tunicates play a very important role to do that. So, there are very important point, I would like to see here, that the drug resistance of the anti-cancers. So anti-cancers molecule, initially, as you know, that tyrosine kinase inhibitor, we thought of that it will abolish almost all the cancer from the world. But soon we find that it has a limitation. It developed resistance within 5 years because of the cancer cell took it's very smart cell. They took a different pathways. Again, we start looking for, and that is why the tunicates are being exploited, terbinafine, plitidepsins, and already there is a drug based on these, apelidine, which is in the market, is coming, and that is working against the drug-resistant breast cancer, human kidney carcinoma, as well as multiple myeloma. Cyanobacteria. Cyanobacteria, this is one organism that most of the marine organisms is very difficult to grow, and directly harvesting from marine sources will disbalance the ecosystem. But the plus point of cyanobacteria is that if we can create an artificial environment, these bacteria can be cultured. Okay, even deep sea bacteria also can be cultured if we can create the environment. So cyanobacteria are, basically initially it was focused that we can produce the toxin from there. Recently, several studies have been demonstrated important biological properties of the compound from the cyanobacteria, and there are already, there are a couple of drugs are there in the market, and a couple of compounds are in the pipeline. I will give an example of that. So these, another example is a very important aprotoxin produced by species Lingibia is potently cytotoxic in human lung cancer, and likewise this cyclophosphamide. And this is also, a very important anti-cancer drug. So as I told you that aprotoxin D, it is, it is one of the important drugs for the human lung cancer cells. Now, this has some sort of even specificity, okay? And, I will come in another important area of the epigenetic alterations, that is not your gene, the gene modification, how functions. And epigenetic modifications are largely used in the disease manifestation and the disease prognosis. So they are also, such as, for example, the histone deacetylase, which actually deacetylates it cut the acetyl group, and thereby it pushed the gene for reparations. So in cancer cells, you will find out, and even some of the bacterial diseases, the deacetylase are very important. And if you can inhibit them, that has a tremendous possibility of new generation drug. Couple of drugs are there, not from the marine sources yet. This is just to, show you, those who are not from the field, that the simple cyanobacteria, what a diversified molecules it can synthesize. And the groups I have highlighted here are the pharmacophores. And you see, these cyanobacteria can synthesize therapeutic peptide to the large complex molecule, including the cyclic molecules. And these are, these are very, very important source of new scaffold, new chemistry, and definitely new generation therapeutics. So next is the marine fungi. So fungi is largely looked for the polyketides, okay, and non-ribosomal peptides, terpenes, polyketide terpene hybrids, the polyketide non-ribosomal peptide hybrids. So these are also the source of rich source of therapeutics used for the antibiotics can be used. Cholesterol lowering agents, that is the lovastatin, as you know, that is actually marine source, and also some of the immunosuppressants. Now, immunosuppression, as we are advancing in our life, as we are advancing in the evolution, we are extending our age. As you grow old, you need more and more these sorts of drugs. Okay, so this is just to give an example. From the fungi sources, microcin is one of them. That has a tremendous possibilities to be an antibiotic, and it is being already used in treating the infection of the urinary tract, nose, throat, and skin. This is one point I would like to say, that I do not know if anybody in this audience are there, those who are unfortunate to see drug resistance UTI, urinary tract infections. And it can happen that all the drugs available in the market, your bacteria which is infected, and the person may not even survive more than 6 weeks. And this is why this is very important area to generate the antibiotics for UTI infections. And this myrcene is one of these compounds which is very, very important. Then the cytospoils. So this is again the cytotoxic molecule, and so this is being now exploited for the human lung carcinoma. One of the important source of drugs as well as vitamins as well as the food for human being is the seaweed. Good part is now country like Japan and some of the countries, those who depends upon in some parts of China, they are now doing the cultivation of seaweeds in the low-lying seas, or even in the seawater, putting into their artificial systems. And there are edible seaweeds. These have a lot of bioactive compound, soluble dietary fibers, proteins, peptides, minerals, vitamins, and that is why and recently it has been found that long time people are consuming a type of seaweeds which can prevent them even to get the cancer. Okay, so very ancient days, seaweeds used to be used for gelling only and thickening the agents in the food, or in the pharmaceutical industry. But recent years, people are using these also for the source of drugs. And one of these example I was very important, and although I have written many of them, I can share these slides. I can send it to NIO for your, if it is necessary for you. This fucodans. So this is a very important compound for the anti-cancer for its anti-cancer property, and its dose dependently inhibit the proliferation and colony formation, both breast cancer and melanoma cells. So the beauty, why I'm mentioning these, that these compounds are not only just cytotoxic, it can actually target the cancer. And the reason is most likely they target this predominant metabolic pathway. The other colorful organs are there in the sponges. The marine sponges produce compounds that affect the targeted disease by different mechanisms. So it can actually do the microtubule stabilization or interaction with the DNA to combat the tumors. DNA binding drugs are the first line therapy, chemotherapy in the world, especially the developing world and the poor economy world, because it is more non-specific. But if DNA binding drugs can be attributed with some sort of a, the specificity, either by double targeting of it or making the hybrid molecule, and getting a new DNA binding drugs will be very important, and marine source could be very important for that. So most bioactive metabolites from sponges proved to be inhibitors for certain enzymes, which often mediates the produce of the mediators of intercellular and intracellular messengers involved in pathogenesis of many diseases, including metabolic disorder. And in these, there are different classes of molecule. One is the lectins. These are basically used for the cell aggregations, and these can induce the cell death. Heteronamine, and this is also could be used as the anti-cancer molecules. Diatoms. Generally, diatoms are rich in saturated and unsaturated fatty acids, esters, acyl lipids, sterols, proteins, and flavonoids. Now, flavonoids, the marine flavonoids are quite a bit different than the flavonoids we have in the fruit. And these flavonoids are basically has much long stability in the cells. These compounds have been reported to exhibit the antioxidant ability. Normally, flavonoids do so, antibacterial activities, and also the anti-cancer activities. This is just to give you the list of a few and different flavonoids and the sterols which comes from the diatoms. So marine natural flora and fauna. We are talking at these to be used either as for the nutrition or for the therapeutics. But, Stephen DeFelici in 1989, he came up the terminology called nutrition and pharmaceuticals. This is called the nutraceuticals. So this will be giving both the health benefit as well as it will help us to fight the diseases. And marine compound are the best source of nutraceuticals. Okay, and if you look at the proteins and peptides from these, like collagen, gelatin, albumin, and this comes from either from the fish, so crustaceans and low-fat fish, and this has many activities besides the antioxidant activity. This can act as an anticoagulant. This can the anti-skin aging activities. As I told you that life is lengthening, so we have to have a smart aging rather than just aging. Okay, and carginin, then chitin, chitosan, and derivatives. Now, these chitins and chitosans, today's world, they are not only using for the gelling agent or edible protective films, it also being used, this chitosan nanoparticle. Chitosan nanoparticle has a tremendous possibility for the drug delivery. And you can actually modify the chitosan nanoparticle with the antibody tagging, with the molecule tagging, and that can be having a big use for that. You all know that omega-3 fatty acids, and in the market you often get the cod liver and tablets and so-called liver oil tablets. But omega-3 fatty acids is a very important nutraceuticals, and this can be actually exploited in a many ways. So some of the phenolic compounds and other pigments which comes from different marine organisms like algae and brown algae and different algae, that fluorotannin, carotenoids, and chlorophylls of different types of chlorophylls, which has not only the vitamin source, it can also be antioxidant and anti-cancer activities. So this we don't need. So marine natural products inspired synthesis of therapeutics. As I was telling in the beginning, that where do we learn? How do we design the molecule? Our knowledge is exhausted, but nature's knowledge is not exhausted. If you see the marine compound, I just showed you the cyanobacteria, but each of the marine compound can be so complex, so beautiful that human being cannot even think and design. But human being has the ability to explore it, to understand the structure, and create that as a need molecule, and use that for the then go for the drug discovery. And many of them has become nowadays, even though it has become very difficult, it is not easy to synthesize, but some of the scaffold, some part of the scaffold has also been become a drug. I will end my talk by giving the examples of few FDA approved drugs from marine sources. And I would like to say that, in our projects, which I told you at the beginning, we are trying to make at least a couple of them affordable for the country. and one from the and 3 different sources. So this is a few examples of the FDA approved drugs. So one is called adrenocorticosteroids. So this is the source is cyanobacteria, and the mode of action, it consists of the chimeric monoclonal antibody. You can target with that and then target the cell membrane protein CD30. The drug induces tumor cell apoptosis by blocking cell cycle progressions as the cell progresses. And this is a more targeted therapy because it can target the CD30 cells. So Trabec, this is 2015, and this is molecule is trabectedin, and it is a C-screte, so it can actually therapeutic for these advanced soft tissue sarcoma. So it blocks the DNA binding of oncogenic transcription factor. Why oncogenic transcription factor? Some same transcription factor, because of the mutation, it forms the fusion protein. This fusion protein will not follow any rules and law of what you expect the cell cycle should do that. And thereby, they become notorious. But if you have a drug which can actually blocks the binding ability of these, new binding ability of these fusion factors, that can do that. And trabectedin is one of them. Halaven, it is a very important drug. It is targeting the therapeutics of breast cancer and liposarcoma. It induces the apoptosis of the cancer cells, prolonged, and it irreversible mitotic inhibitors. Okay, but it has a huge side effect because of its, the irreversible mitotic inhibition. However, this has been approved. Another is Cytosar. So this is basically a new generation therapeutics. It's old generation therapeutics for leukemia. Leukemia generally, treated, as most of you know, that it is still, the cytotoxic drugs like, doxorubicin and other cisplatins. These are very much being used, but the sponge-derived Cytosar is a very important source of the leukemia to be, targeted therapy for leukemia, because arabinose sugar sterically hinder the rotation of the molecule within the DNA, and thereby it stops the DNA replication, and thereby it becomes a very important therapeutic molecule. So, so the Feridac and apelidin. So one of them and both of them are very important drug for this multiple myeloma. Chronic pain. So you see, pain drugs are very important because, again, as life is extending, the cases of cancer is increasing. Cases of metabolic disorder is increasing. Because of this, people live longer, they keep accumulating more and more mutations. So the, and the pain is another thing, the different types of pain. Even the cancer therapeutics can bring a lot of pain to the patients. So the painkillers are very important, new generation painkillers, which will be actually not harming us too much. Any painkiller will harm us, but will give the new, the more targeted pains, different types of targeted pains is very important. So the gicotinide, it is a synthetic peptide analog of omega conotoxin, and it's a it's an analgesic molecule, as I said. It blocks the calcium influx to the calcium channels that are present in the spinal cord, thus preventing the afferent conduction of the nerve signals and being used for the nerve pain. Viral diseases, there are many compounds are being exploited, and especially the corona has made us, but some of the drugs are already in the market. This is, and CSIR, very interestingly, has a viral mission. As I understand, at this moment, it is in a few institutions along with CDRI, it is going on very well. Then hypertriglyceridemia. So this is, many drugs are available in the market, but some drugs work for someone and some drug doesn't work for someone, because the cause of the the hypertriglycemia for different persons are different. And that is why, like hypertension, we need many varieties of drug targeting many different newly discovered targets. And Lovaza-2 Vesapa, these are these are the new drugs which come from the fish oil, and it is being a very important source. Many compounds for antiviral and anti-cancer are in the different phases of clinical trial. And the sources are from cyanobacteria, mollusks, and so these are just few list I have given. The phase 1, different phase 1 drugs which are in advanced stage, different phase 2 drugs, and among them I must, biostatin, which is looks like is a very promising for the Alzheimer's disease, and it is being now taking in advanced stage also. And of course, newer and newer solid tumor targeted things are coming up. In the phase 3 clinical trial, so this is one important thing. There are few drugs which are, as I told you, that very important for the chronic pain. And the sterotoxin, so this is a chronic pain drug, is in the phase 3 clinical trial. And there are antibacterial, there are non-small cell lung carcinomas, pancreatic carcinomas, melanoma, lymphoma, multiple myeloma. 2 important drugs hopefully are in the pipeline. And tunicate source, this pelidipepsin, is also in the phase 3 clinical trial for the coronaviruses. And this has a good possibility to be incorporated in the therapeutic regime of broad spectrum antiviral address. So, as I told you, that as we are going along, we are more and more understanding that it is not your gene, it is the gene modifications are very important for your life phenomenon as well as a disease response. Thus, the epigenetic target like different histone modifying enzymes, DNA methylations, the microRNAs, these are all become a big therapeutic target. Marine sources are being exploited. Largely so far, the marine sources have been exploited for the DNA methyltransferase inhibitors and histone deacetylase inhibitors. And these are few compounds which I have listed here, that these are being tested, and some of them is in the advanced stages of the clinical trial. So what I said to you so far, there are, you don't need to read these slides, but I can summarize by 3 points. One is the diverse source of different scaffold in the marine sources are very important source for our knowledge of chemistry to design the drugs, to combat the new resistance, to combat the new diseases and the new viruses. Besides, marine source can be a very good source of nutraceuticals, which will give the therapeutic effect as well as the nutrition. Third, marine source, if we can exploit properly, if we can design the culture, it can be very good source of nutritious food. Especially country like India should exploit more and more the marine source as a food through the artificial cultures. I'm sure, I'm confident that institute like Triple IM Jammu, institute like NIPERS, and institute like CDRI will be guided and inspired by CSI NIO, and to make as Government of India is planning, if I understand correct, a large program, mega program on the marine sources, both for therapeutics and food. Thank you very much for giving me the opportunity. I thank again Professor Sunil Singh for his kindness to give me this opportunity to talk to you and brush my knowledge as well. Thank you very much.
Thank you, sir, for your very interesting talk and for emphasizing the relevance of this event and the significance at looking at marine resources. Moreover, you have highlighted important issues like therapeutics, drug resistance, seaweeds as a source of bioactive compounds. We will take one or two questions. If anyone from the audience or the delegates wish to ask Professor Kundu, you can raise your hands. If not, I will ask, sir, with the
Yes.
increasing trend in drug resistance, from your perspective, what could be a way out besides finding out new drugs? Is there a way out? Or do you think, with respect to synthetic biology, it will get an upper hand over,
So you see, if you say the way out, there are two different ways immediately we can think. First of all, some drug resistance are man-made, especially the antibiotics. Antibiotic resistance are because of indiscriminate use of antibiotics for no reason. Second thing is that the food contamination with the antibiotics. So that is, that is why the drug discovery, we also looked very carefully the veterinary medicine. Okay. Because, the lots of sorts of the unwanted antibiotics are from there. Second thing is, the drug resistance for the cancer. Drug resistance for the cancer is difficult to stop by our wish. But as we understand the fundamental pathobiology of the drug resistance, most of the time the drug resistance is appearing because of certain associate epigenetic modification upon using the same drug for a long time. Cancer cell is a very smart. Antiviral and TB resistance, TB drug resistance. This is again, the TB drug resistance, if you see, this comes around that the same reason is that, the half treatment, no treatment, or co-op treatment, and giving the bacteria to take those advantages. The other drug resistance, what we are having, these are difficult to resist, but the way out is to discover the mechanism and thereby discover the new target, and marine source could be new target and new therapeutics. Marine source could be very, very inspiring for us.
Thank you, sir. It was a privilege to have you on this event. Thank you.
Thank you.
To follow, we have an overview of marine bioprospecting R&D program at CSIR NIO by Dr. Narsim Thakur, Senior Principal Scientist at CSIR NIO, the event coordinator, and is also leading the multi-institutional bioprospecting project, which is aimed at developing marine-derived products and processes in collaboration with industrial partners. Over to you, Dr. Thakur.
Yes, good morning. My slides are visible.
Yes, it's visible.
Okay. Yes, thank you. Thank you. Yeah. First of all, thank you very much, Professor Kundu, for giving us an overview of current progress in this particular field, and more especially for highlighting the importance of this particular topic. As you said correctly, NCE, that is new chemical entities, are required for us, as nature is just showing us a cue or structure of the molecule which we can take further by synthetic route. So here in brief, I'm going to give you overview of the marine biotechnology program or marine bioprospecting program at CSIR National Institute of Oceanography. CSIR NIO headquarters is in Goa, and we have 3 regional centers. And this is a premier multidisciplinary organization working in various fields, right from physics, chemistry, biology, geology, geophysics of the ocean, and also marine instrumentation, ocean engineering, and marine biotechnology. So these are our theme areas as far as marine bioprospecting is concerned. That is therapeutic molecules. Those are bioactive secondary molecules from various marine organisms. Then functional foods and supplements, natural pigments, especially from microalgae, hydrocolloids, those are the products from algae, and microbial enzymes for various industrial applications. And this is an area that is marine living resources. The rich marine living resources are available, and these marine living resources are very much useful for the applications in various industrial sectors. The marine living resources are marine invertebrates from different depths, then cold sea and deep sea biota, microalgae and macroalgae, including seaweeds, microbes associated with marine organisms. As we were going through the lecture of Professor Kundu, he was highlighting the symbiotic association and the importance of that in drug discovery. So there are a lot of microbes which are associated with marine invertebrates, and these microbes are producing valuable bioactive metabolites. And then extremophiles. So these are our marine resources, living resources, where our group is working. And our strength is that we have a group of almost 25 scientists involved in marine biotechnology research. Then more than 2,200 publications related to marine biotechnology since last 10 years. This particular point I would like to highlight because this is a fundamental research in the field of marine biotechnology. Maybe not many drugs or bioactive molecules have been delivered to the industry, but this is a basic or fundamental knowledge or the expertise we have established since last 10 or more than 10 years. Then more than 50 national and international patents have been filed and granted in this particular area. Then marine microbial culture collection. This is a very unique facility which we have. So you can see more than 2,000 unique microbial cultures are there with us. These cultures are from different depths and different ecological niches. Then marine natural product repository, where we have more than 1,000 metabolites with us, and this continuously we are adding metabolites in this particular marine natural product repository. So this is our strength, and we have state-of-art laboratories including marine natural product chemistry, molecular biology, microbiology including fermentation, and so on and so forth. So very unique facility which National Institute of Oceanography is having. We have two research vessels. One is Sindhu Sadhana, where endurance is 45 days, and Sindhu Sankalp. So these two research vessels we are using for sampling purpose. And recently we have completed a cruise, a very long cruise of 90 days in Indian Ocean, and collected a lot of samples from various depths. And on board, definitely there are some sampling equipments to collect the samples from different depths. And on board, there are laboratories to process the samples immediately on board, and then we can bring these samples to the main laboratory at headquarters. So this is our strength. Apart from, we have a diving group. Diving group can collect the samples from coral reef and other areas. Then something about the technologies, some of the technologies which we have licensed or commercialized, and some technologies are ready for licensing. First one is a DHA, that is omega-3 fatty acid. Just now, Professor Kundu has highlighted the importance of this particular omega-3 fatty acid from marine sources. It is from Throstochytid strain. This particular technology was jointly developed under NIMITLI program by CSIR NIO, CSIR IIIM, and CSIR IICT. And this is licensed to the company. Then decolorization and detoxification of molasses paint wash using marine fungi. This technology is available with us. A process for production of low temperature active alkaline protease from deep sea fungus. Very unique enzyme is there with us. Novel 6-terpene oxides as perfuming and flavoring agent. And finally, the melanin from sponge-associated bacteria. This particular technology is also licensed, and we are working with the industry for scale-up. There is an ongoing program in this particular field that is synergizing marine chemical ecology with bioprospecting. Just now we heard that these organisms are producing bioactive metabolites for their own purpose in the system. So we have to understand this chemical ecology, which is including the metabolites which are used for defense and communication purpose. So in this particular project, our aim is to understand the chemical ecology and translating this particular knowledge for developing products and processes for industrial and societal application. So this Bioprosper project is funded by CSR under NCP category, that is niche creating project. Nodal laboratory is CSR NIO, and there are 5 participating laboratories in this: IIM, IMTech, NCL, CGCRI, and CFTRI. So CGCRI, we are working on marine-derived biomaterial, and CFTRI for food-related research. So this project duration is 3 years. But during this project, we have developed some leads. Of course, first lead is not from this particular project, but we are working on this particular lead in this project. That is a cyclic peptide, novel cyclic peptide, having application as an antibiotic against gram-positive strains. And this particular antibiotic is effective against hospital-acquired infections like MRSA and VRE. And we are working on this particular antibiotic in collaboration with industry. Then collagen from jellyfish, water-soluble chitosan derivatives for biomedical application, algal oligosaccharides, ulvan and porphyrin as prebiotics, beta-carotene and fucoxanthin pigments from microalgae, amphiphore live feed, and finally 3D printed material for immobilization of bacteria. So these are some of the leads where we would like to have industrial collaboration, and from this meeting we are expecting some of the potential industries interested in these leads can contact us. The objective of this iConnect event is that awareness about the marine biotechnology research going on in NIO, that we would like to spread this awareness. Then most importantly, networking with potential industries, then handholding with industries for scaling up the potential leads which just now I have shown, and finally to formulate joint R&D programs in very specific areas. So agenda of this meeting is that, followed by my overview lecture, there will be plenary talks by industry leaders, and there will be a panel discussion between scientists, industries, and line ministries. Thank you so much.
Thank you, Dr. Thakur, for giving an overview on marine bioprospecting at CSIR NIO and also on the objective of iConnect event. I thank all our distinguished speakers for their perspectives on this event. Thank you. Next, we will have the plenary talks. Each speaker has 10 minutes. Those who wish to pose questions after the talk may type your questions to the speaker in the chat box, some of which will be answered after the talk, while some questions due to time constraint will be responded by email. So also write your email address in the chat box as you type your query. I welcome the first speaker, Dr. R. Dinesh Kumar, Senior Scientist, CSIR, CSMCRI, who will speak on marine biotechnology research at Central Salt and Marine Chemical Research Institute. Dr. Ravindran, Principal Scientist, NIO, will elaborate on the speaker.
Thank you, Dr. Judith. I'm glad to introduce Mr. Dr. Dinesh Kumar Ramalingam, from Senior Scientist from CSIR CSM CRI. He has obtained his PhD degree from Biochemical Engineering subject from IIT Kharagpur. He also worked as an assistant professor under Bioengineering Division from SRM University, Chennai. His research skills and interests involve an interfacial role between chemical engineering and biology. His expertise in cultivating microalgae used closed photobioreactors and raceway ponds, and also in developing cost and energy efficient downstream process. His current research interests include designing and optimizing upstream and downstream processes for improved manufacturing of bioproducts that benefits our society in the sectors of healthcare, energy, and environment. With this short introduction, I just request Dinesh Kumar to deliver his speech, please.
Dr. Dinesh Kumar, over to you. Kindly unmute.
Yeah. Thank you for your introduction. Hope I'm audible now.
Yes, you are.
Yeah, thank you. And my slides are visible as well?
Yes, your slides are visible.
Thank you. Yeah. In this talk, I'm going to cover up the significant findings that we got from marine biotechnology research at CSIR, CSM, CRI. In this talk, actually, there are 3 technologies that is developed by some of our scientists would be discussed. The first one would be by Dr. Soumya Haldar and his team. This is about, uh, this is about AquaBoost, which is an indigenous symbiotic product, especially developed in view of the issues that is faced in aquaculture farm industries. As we are aware that in aquaculture farm industries, there is a hydrogen sulfate production problem due to which there would be a blackening of aquaculture sediments. So, and there are like consequent effects like reduction in, the growth of the shrimps and fishes. So to overcome these issues, there are market products available in the existing market, but although physical chemical methods and some probiotics are available, but they are not indigenous in nature, and they have some fitness issues like with respect to ecology and high cost. To overcome that, Dr. Haldar Group has successfully developed AquaBoost product, which is a combination of a prebiotic and probiotic. The prebiotic is actually from a plant origin product like vinasse. As we know, it's a sugarcane byproduct, sugarcane based ethanol production industry byproduct. This vinasse has been, the removal of potassium has been done using a separate technology, and this has been used as a prebiotic one. And there are 3 consortium which has been isolated from mangrove forest. They are like Enterobacters, Pseudomonas, and Cytobacillus strain. So the formulations have been successfully developed in this ratio of 1:1 ratio along with the prebiotic. The striking features, or the salient features of this particular product is that this consortium can tolerate a wide range of pH and salinity, and it is safe against the model like Otinia. So it has, since the water aquaculture pond water would be released successfully, so it has been tested against this sensitiveness towards the commercially and relevant antibiotics, so that it will be safe to use. And also a lot of the products already existing in the market, already existing in the market, have the issue of storage problems. But the present technology given us a very good stability in the room temperature for about 6 to 7 months, and there is no significant change in the viable count as well. So due to which this product, it's kind of robust, and it can, not only withstand the wide range of pH and salinity, it can drastically reduce hydrogen sulfate production in the aquaculture pond, and it can reduce the load of Vibrio. Right now, the technology has been patented, and the present state of the technology is that some third-party field trial is actually undergoing in the states of West Bengal, Orissa, and Gujarat. Once it is success with vis-a-vis the commercially available product, and this would be taken up by the industries, aquaculture industries as well. Any queries related to this particular product AquaBoost can be posted to Dr. Haldar, and his email ID is given here. Thank you. And then the next one is about the production of GLA. We call like gamma linolenic acid from microalgae, marine microalgae. Basically, this technology is developed by Dr. Saurish Bhattacharya. The idea is that to use the marine microalgae chlorella variabilis, which is actually he has underwent some medium optimization, and the yield of GLA is about 5% with respect to dry biomass. And the de-oil biomass have been utilized for the production of some marine bacteria like Bacillus licheniformis, and that in turn produces epsilon polylysine, which is used as a natural preservative. And he has done some cost economics, which comes around, the production of GLA is about 890 rupees per gram, whereas epsilon polylysine is about 11 rupees per gram, and protein powder is about 400 rupees per gram. So this technology is also available for technology transfer. And there are any questions related to this GLA production through biorefinery approach from microalgae, maybe ask from Dr. Saurish Bhattacharya, and his email ID has been also been given. The third technology, which is at the TRL level of 3, is actually developed by me. Basically, it talks about the production of lutein from marine microalgae Dunalella, and it's a very well known nutraceutical property that is for eye health. Basically, under R&D scale, the medium and process conditions have been optimized, and it has been successfully validated in outdoor open conditions. As you can see, in we have a terrace facility, or in that, say each tank is about 400 liter capacity, and also it has been tested in the raceway pond system here, which we have it to the pot culture, and it is about each raceway pond is about 4,000 liter capacity. So the upstream process have been successfully validated in large conditions. The productivity of 25 to 28 gram per meter square per day, which is equivalent to 2.5 ton to 3 tons per hectare, have been achieved in open conditions, and that is a conducive season like April to May month. In this year, and we are actually kind of taking up it throughout the year, cultivating this microalgae throughout the year to understand the seasonal variation and the contamination issues and how to rectify it. So whereas in the downstream, we have developed a integrated downstream process which can actually separate lutein and phytol concurrently, because what we understand, producing a single product from microalgae would be less economically attractive. So we want to target another high value product like phytol, which is actually a component of chlorophyll, and it has some industrial applications, and it has some market demand as well. So the developed downstream process has given purity of lutein about 84%, and phytol is about 63%. And this is at technology level 3. And whereas in the upstream, we are trying to move towards the TRL level between 4 to 5. Basically, the idea is to, the basic USP of this particular technology is that the complete biomass valorization for concurrent separation of two high value products like lutein and phytol, along with achieving the purity of about 80%. So that is about this particular technology. Any questions related to this particular, and this particular technology needs some intervention in the downstream process extract, especially in the harvesting and cell destruction part, where we found there are a lot of energy has been consumed. So we are working on developing integrated unit operations to achieve this harvesting and cell destruction as economic as possible, so that the whole process could be economically viable in industry point of view. Then this technology would be ready for this process would be available for technology transfer. So with this, I like to thank. There are other technologies as well, but this is in view of E3OAA category. We would do work on seaweeds, and that would be taken into separate IConnect events. So I've restricted my talk within the 3 technologies here. And thank you very much for the opportunity.
Thank you for your informative talk on marine biotechnology initiatives. You were kind enough to provide the email contacts. So all the audience, if you all have any questions regarding the presentation, you can contact by email the respective person. But I do have one question for you. So as you are into so much of marine biotechnology research, do you interact with industry to scale up your technology?
Yes, we are in constant touch. The first product, Aquabus is proposed. Actually, Dr. Haldar is in touch with many aquaculture farm industries. And actually, the third large-scale field trials are actually going on in the states of West Bengal, Orissa, and Gujarat to understand, different, climatic variations of how this product works. And also the last technology which I proposed, like that is lutein, and for which we are in contact with some industries like Seagrass Tech Private Limited, Puducherry, Pondicherry, and also Sai Ishwar Biosciences in Chennai. So we are in touch with the industries as well.
Thank you, Dr. Dinesh Kumar.
Thank you.
A warm welcome to Dr. Rahul Varke, Director, High Media Laboratories Private Limited, who will throw light on marine hydrocolloids. Dr. Raki, Principal Scientist, NIO, will say a few words about Dr. Varke.
It's my pleasure to introduce Dr. Valke to all of you.
Dr. Varke, are we audible?
Yes. Can you hear me?
Yes, sir.
Yes, we can hear you. Dr. Valke is a well-known scientist and expert in international business marketing and Director of Research and Development Wing of Microbiology Business at High Media Laboratory Private Limited. He completed his master's as well as his doctoral research in microbiology from Institute of Chemical Technology, Mumbai. He continued his research career in Walter Reed Army Institute of Research, Maryland, USA. Since his return to India in the year 2004, Dr. Rahul has developed an entire range of animal-free microbiological media which has found application in vaccine and biopharma industries at a global level. He has been instrumental in introducing a fully automated plant for manufacturing of ready prepared media in India. Dr. Rahul Varke represents Bureau of Indian Standards as a technical expert in food microbiology at the ISO Technical Committee for Food Testing. He has been a life member of American Society of Microbiology. With this brief introduction, now I request Dr. Rahul Varke to share his presentation on marine hydrochlorides with us. Over to Dr. Rahul Varke.
Yeah, thank you, madam. Thank you so much. I'm just sharing my presentation. Hello. I'm having some. Girish, can you share? I'm having some trouble sharing.
Yeah, I will just share.
Yeah.
Can you all see?
Yes.
Okay, great. I
Yeah,
think
Girish, next slide. Yeah. 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 So, thank you everybody, and thank you so much for inviting me for giving this presentation. And we really appreciate the initiative taken by NIO and the entire team, because marine hydrocolloids has become a big challenge in India, and we've been facing this problem since 1974, 75, since high media started. And we are the biggest consumers of hydrocolloids, I think, right now, as far as for microbiological applications is concerned in India, and probably the second or the third largest in the world. So we've used quite a bit of agar, and we use few hundred tons of agar every year. And it has always been a nightmare as a manufacturer of culture media and as an exporter of culture media globally to get good quality of seaweed to make agar. So what is really important is we need to think about the sustainability of the hydrocolloids in our country. So as far as are we really as strong as we think in hydrocolloids? I don't think we are, because we are just catering to one or two commercial products, and that too on a very pilot scale. We require much more uh, what you say, involvement by both the industry, academia, and also by the government in terms of policy making for uh, making this industry viable in India. Girish, next slide, please. So uh, a big chunk of our supply of hydrocolloid is coming from China and other global suppliers. We need to break this comfort. And I guess the last 2 years of pandemic was one of an eye opener for a lot of industries and academia in India, when the supply chains kind of dried up in from most parts of the world. and everybody started looking internally that how and where we can close these gaps of importation. So we need to really get over this comfort zone and move on. Next slide, please. Thanks. So the seaweed market, if you look at the seaweed market globally, it is growing at a CAGR of 9.1%. It is estimated to be 23.2 billion. So these are numbers for the entire hydrocolloid market, out of which if we see the types of seaweeds are the red seaweed, brown seaweed, and the green seaweed. And the most common application at this point definitely is food and beverage. Then extraction of hydrocolloids, and then agriculture and others. So by geography, if you see, the largest share of hydrocolloids is, I'm sorry about this slide, about this picture, but let's look at the application. And if we see the applications are mostly in the food and beverage industry right now. Nobody has talked about our applications in biotechnology. So if you ask me what are the applications in industry, apart from the food industry, we use it in microbiology media. Microbiology media is one of the biggest go-to the raw material is been agar. And second, very big applications of agar has emerged in downstream processing in the biotech industry, especially where they are making all the monoclonal antibodies, where they use a lot of agarose beads. And the agarose beads are becoming so expensive because the downstream processing of many biomolecules, proteins, you need very specialty agarose beads, and these all come from hydrocolloids, especially from agar like geladium. Next slide, please. So let us, at this point, what is our association with the hydrocolloid business? So first is alginates. We don't make any alginates. We source alginates, but that is a big business. We do source and we purify it and grade it and sell it. Then we have agar. Agar is not only consumed internally to make our dehydrated culture media, it is also used to make a lot of plates. It is also used in various forms of prepared formats of medias. Then we also use a lot of carrageenan and then agarose. As I briefly mentioned, the various applications of agarose in not only in the biotechnology but also in the research and fields of proteomics. Next slide, please. So here is so basically there are two major the biggest chunk of agar is basically comes from seaweed, which is either geladium or crassularia. The geladium species is a very good species as far as applications in microbiological agar are required, and this requirement comes from two basic aspects of geladium's gel agar, which is it is extremely clear. The gel which is formed is very clear, and second, it has very low gelling strength, which helps addition of supplements into the agar like blood or antibiotics, which is most commonly used. And third, another critical parameter of geladium, which is useful for microbiologists, is its pH range. So the pH, the gel is stable at a very broad spectrum of pH, which helps in designing various microbiological applications, right from very low pH to grow fungi and candida to very high pH used to growing other kinds of bacteria. Next slide, Girish. So I'll briefly go through the chemistry of agar, and I think Dr. Dinesh already covered most of it in his previous talk. But agar are basically gel gel-serving polysaccharide extracts from agarophyte, and they are a complex mixture of polysaccharides composed of two major fractions, agarose and agaropectin. So agarose are gelling fractions in a neutral linear molecule essential free of sulfates consists of chains of repeating alternate units of beta 1,3 linked beta galactose and alpha 1,4 linked alpha 3,6 anhydro L-galactose. Agaropectins and non-gelling fractions is a sulfate polysaccharide, 3 to 10% sulfates composed to agarose and verifying percentage of ester sulfate, D-glucuronic acid, and small amounts of pyruvic acid. So, I mean, I will not go through this in detail because we know that agar is basically composed of two, is agaropectin and so here is a typical hydrocolloid of how is a hydrocolloid, the chemistry of the hydrocolloid of an agar. Next slide, Ganesh. So this is a very important slide, and it needs to be taken seriously. And we kind of were shocked a few years ago. I mean, almost 2015, there was a big publication which came out in Nature, and Nature had to publish a paper. If you see this on the top left hand side, there is this girl walking over the seaweed. So this is a picture from Nature 2015, where they say that lab supply agar runs low. So in 2015, the Nature had to publish this because what was happening is globally there were a lot of restrictions which were coming in on harvesting of seaweed. So, because the harvesting of seaweed was causing a lot of environmental damages, the way the people were harvesting the seaweed from the sea was not sustainable. B, the European government had lots of regulations in terms of the fishing because people were overfishing in the same way they were over harvesting the seaweed. So that was that, and a lot of seaweed which was being harvested was not processed properly, and there were many challenges. So which was leading to a huge which was leading to a huge reduction in the supply of agar. So this kind of created a big shortage, and this shortage is kind of increased the price of agar by at least fourfold in the last 1 decade. So the seaweed agar which we were buying at X is now almost 4x, and it again shot up after the pandemic. So which means that the only way this can, if this has to be sustainable, is that by doing uh, uh, cultivating it. So High Media has tied up with CMCRI and the team from CMCRI Bhavnagar, where we decided to cultivate the Glacilaria species, and this was done to see if Glacilaria can be done in India, and if that, and then we were thinking of doing Gelidelia because Gelidelia needs specific temperature requirements. So we thought, let us start with Glacilaria and see how we can sustainably do it. So the first project was done in near the coastline of Diu, near the coastline of between the Bhavnagar and Diu. And this is how the first harvest which came out was. This was cultivated by CMCRI and their team. And you can see that this cultivation was very good. The purity of the seaweed is fantastic, and the color and uh, uh, the production of the seaweed was very good. After that, we did downstream processing. Next slide, Girish.
Yeah.
And if you can see in that, so the downstream and facility which we set up in our facility to further this, and we were able to manage to get a good quality of agar out of this collaboration. And unfortunately, because of the pandemic, we had to take a back seat, but we are now back again. So the two agar samples received from the factory, they showed very good results as compared to the ones which we were importing commercially from other parts of the world. So if you see the gelling strength, the pHs are in range. The gelling strength is pretty good. The gelling temperature also is very satisfactory. So in short, we were able to, for the first time, we were able to cultivate and produce an agar. This was, of course, at a pilot scale, and we are still in. Now we want to go ahead because we feel that it's a sustainable project. Also for the fishermen of the regions of Gujarat, where they can culture this seaweed, and we can take this to a different level. So, as I mentioned, we are fine-tuning this project, downstream processing with respect to specifications of ongoing with CMCRI and our team. So, next slide, Gireesh. Yeah. So with this, we feel that we are doing our bit to be a part of this sustainability because we need this raw material, and we would be and we are probably emerging as probably one of the top 2 or 3 suppliers of culture media in the world. So we will be consuming a couple of 100 tons of this seaweed in the next coming years, and we already are actually. And so we think that India should be able to produce this, and I don't see any reason why we cannot, because we have the technology, and we, with the support of CSR and CMCRI, we definitely be able to make India one of the top producers of agar in the world. So thank you. With that, I can take any questions if there's any.
I really don't have question, but I would like to comment something. First of all, a big congratulations to CMCRI people that for doing this work and connecting high media. And my comment is, Government of India is thinking to have a national marine mission, okay? And I think one of the vertical should be this agar. And I will, whenever the thing comes into the picture, we may need to sit down together. And one of the vertical we should put it agar. It has many other reasons to be more than the lab shortage to supply, but it's a fantastic, congratulations, a big congratulations. This really looks very promising, very much needed at this moment, and I highly appreciate that. So, Sunil is coming in. So I would say that whenever this national marine mission, whatever people are talking of, and so whenever this comes, this one of the vertical we should include, and we can see that around these what kind of programs we can put it, and how many different kinds of scientists we can involve in that, including the industry. Thank you.
Sir, we would appreciate, and congratulations to High Media also, because we've also been a part of
Absolutely.
Thank you, sir, because our team has really worked hard. Of course, credit goes to CMCRI, and it's been a very fantastic start.
In fact, Rahul, this could be an example. The similar example could be in the same way that we do the large production of a specific cyanobacteria. We have done we have just initiated a little procedure on that in the FCM Center for Marine Therapeutics program, which is yet to be approved. But this is very good example. This is very good example. This is very inspiring.
Thank you, sir.
You are not audible. We are not being able to hear. No, not audible. NIO, you are not audible.
Okay. Am I audible now?
Yes, now you are.
Excellent. Thank you, Dr. Worke and Dr. Mahajan. I think your proactive role has really helped all of us, more specifically CSM CRI, to position itself. Now, my question to you is, yes, while this national mission on the algae will come up, it will be prudent if we, in the scientific fraternity, including, experts like Professor Kundu, we have an understanding as to the requirement, essential interventions that you seek, be it agar, be it any other product, so that when these missions actually roll out, we have specific projects and specific research teams identified to take them on. So one of the suggestions or requests that I would like to make to High Media and companies like High Media, if you could share with us the wish list that you have currently, maybe import substitute, maybe process improvement, whichever way it is. So that will also give us an idea as to these are the areas that are sought, and therefore under those missions or independently through CSR also, we could take up projects which would address some of these issues. Thank you.
So this is, this is a very, very important output would be from this meeting. I will request Professor Sunil Singh that, if you select, 10 to 15 potential scientists and industry and as well as a few people, and just call, if possible, an in-person meeting in NIO, and we can fix, we can rather brainstorm that this could be the request for the national mission, and that can go to the directly to PM. Actually, PM, Prime Minister himself has encouraged that national marine mission in different forum, and we can be proactive to send it these through maybe DOP and other industry organizations, and NIO can lead this meeting. I would request Professor Sunil Singh, and then we can incorporate few of that, and that could be a very good output from our today's discussion meeting.
Yeah, thank you very much, Kundu Saab. I think this is a very good suggestion, and we are going to do that. I think we'll have a small meeting, brainstorming, and outcome of that will be sent as you have suggested. So I think we need to be a little bit proactive.
Yes.
And I will request Thakurji to take up this.
And I would request that let us do these from CSIR, NIPER, one or two DST Institute. So let CSIR lead it, but the other people also be there.
Yeah. So that should not be a problem because we should do together.
Yeah.
We should not fragment our efforts. So I think we will be doing that.
Because national mission will be together, this one.
Yes, sure. So I think when we are doing this small brainstorming, we will involve all the small or big institutions, and then we will propose all the things.
Very good.
Yeah, so we'll do it as soon as possible, Thakurji.
Thank you.
Yeah.
Thank you, Professor Kundu, for your suggestions. Thank you, Dr. Warke, for highlighting the importance of having a sustainable system and also the need of the involvement of academia, industry, and government. I now welcome Dr. Santanu Dasgupta, Senior Vice President, Head of Synthetic Biology and Algae to Oil R&D, Reliance Industries Limited, who will talk about the products from marine algae. Dr. Catherine, Principal Scientist, NIO, will introduce the speaker.
Good morning, and it's my immense pleasure to introduce Dr. Shantanu Das Gupta. He has over 30 years of experience in this field in the area of biotechnology, and he's still an active researcher, and he's also leading very key scientific and advisory roles for the Government of India and many academic institutes. He started his research career at Bose Institute, Calcutta, from where he earned his PhD degree in molecular biology, and he then completed his postdoctoral research from University of Kentucky, USA. And after that, he has led many technical and leadership roles as a director, regulatory policy, and scientific affairs at Monsanto Company at St. Louis. After that, he is playing an important and biotech outreach program and regulatory policy for biotech trades in India. He is associated with several international bodies, and he is the member of the Global Council of Synthetic Biology World Economic Forum. Presently, he is a Senate member of various IITs, including Allahabad, Guwahati, and Mumbai. He is also in the Scientific Advisory Board and the Governing Council of Punjab University, University of Kalyani, and also is in the panel of Advisory Committee at CFTRI, Mysore, and Institute of Biomedical Genomics. He is an active researcher, and he's recently also published papers on the use of macroalgae against coronavirus. And his major contribution has been in the field of biofuels, food, feed, nutrition, and biomaterials through synthetic biology. He has over 100 publications in several international journals, and he has also many patents to his credit. And he has tremendously contributed for the traits for developing pathogen resistance in plants, drought tolerance through gene expression, and improved algae, and for the production of high value chemicals. We are very happy to have you, sir, in this panel. Thank you very much.
Thank you very much. And good morning, everyone. Hello. Hope you can hear me.
Yes, sir, we can hear you.
And you can see my slide as well.
Yes, sir.
So thanks for offering your valuable 10 minutes to me today. So I'll not take much time. Very quickly, I'll give an overview. And I'll not talk about, I wrote algae, but it's basically I meant microalgae, and not freshwater microalgae. It's ocean microalgae only, marine microalgae. And I don't have to tell you about the environmental catastrophes, what's going on because of many of the unsustainable things what we do today, and we are doing for hundreds and thousands of years, including the way we do our agriculture. So it's very important, and people already emphasized the need of us focusing on blue manufacturing for sustainability. This is something our chairman told few weeks back actually. So what he said that we do not have a planet B. So whatever we need to do, and we need to do for our future generation, we have to do it today. And we need to bring sustainability everywhere at all sphere of our life. This is the kind of slide you probably have seen hundreds of times by many different people. So I will not go through it, but what I will tell that see, today's world is challenged with ever increasing population and resulting for demand for more food, more water, more energy, better nutrition, and better environment. Now, more biomass produced, I'm talking about biomass now, okay? And more biomass produced in an innovative and sustainable way can solve many of these challenges, what is listed in this slide. And we chose algae and microalgae. And why? Algae offers, and almost all of you would know that algae offers the maximum potential to exploit the most important chemical reaction of this earth and provide maximum possible biomass compared to any land-based crops. You can see the graph on the left-hand side, and I'll not go through the slides. And of course, microalgae offers diverse opportunities, and you all know again, I mean, starting from fuel to personal care to food and feed and materials, and we are in fact interested, and we are working on most of these areas. Okay, application of microalgae to all these areas, what I show in this slide, and I'll give you just 2-3 examples. What we do. But before I do that, I wanted to tell you little history of our algae work. And we started about a decade back, and we have established an end-to-end algae technology capability and large-scale cultivation. Our largest pond size is 2.5 acre, and we are continuously growing algae for more than 4 years. And we have created a large and diverse repository of algae, microalgae, and stage-gated screening procedures for resilient and productive strains. Why I'm emphasizing on the resiliency, as many of you working on algae, you would appreciate the fact that it's very easy to work in the lab with algae, but when you take them outside to get a resilient microalgae working at your backyard is challenge number one. Challenge number two is, of course, the productivity. And we started from high throughput selection in the lab, followed by screening and optimization at different scales. We start with 1 square meter pond up to 2.5 acre pond, as I told you. And ultimately, we select a handful of strains, what we are able to grow at a large scale for a long time with reasonable productivity. As it doesn't matter whatever product you do with the algae, the first and foremost thing is you should be able to grow algae at your backyard. Your backyard could be near an ocean, but that's fine. So some quick example. One of the things we're really interested and we're working. We'll be probably commercializing this in I don't know, maybe 1 to 2 years from now. We call it Reliance protein or R protein from microalgae. And this protein has many different applications we are showing, but one of the applications we're very interested and we are working it. We've already made the prototype for meat and fish alternative. And in fact, ocean and microalgae, if you think about today's agriculture, it's not sustainable. We use like 70% of fresh water for agriculture. So you need to have alternate source of food. And our protein, this protein from microalgae in terms of its quality, digestibility, presence of essential amino acids, this is one of the best source of protein, and you can use it for many different purposes. And it's as good as whey protein, and it's much better than soy protein. This is just we are showing that we made a prototype of meat. Okay, you could see that figure here, and it shows that how do you do the extrusion to produce alternate meat using algae. And people spoke about agar. We just spoke about polysaccharides, and another thing what we are very interested in, we are working on as a material is nanocellulose, that is also from our microalgae. And then algae as a feed. So what we are doing is we are doing a complete cycle. It's like the biomanufacturing is you grow algae, you make protein, you make your materials, you make oil, and also take the leftover biomass and you use it for feed. And we have tested for all kind of feed. We have tested for fish, for shrimp, for poultry, for pet food. So it works absolutely well, and compared to any other common feeds available in the market. This is an example of algae to oil, and as I was telling you, we have established this process, and now in microalgae cultivation and processing, we are one of the best in the world. Okay, even if I compare with the big major oil companies, I think we are. I don't want to name, but we are much ahead of any of them, and you could see the algae biocrude on the right hand side. Dr. Thakur asked me that if I can tell a little bit that if we have to make those gaps little close, industry and academia, what could be the expectations? I think my expectations are pretty simple. One is I would not say it's void, though it's written it's void in academic curriculum. I think in many of the academic institutes started teaching those courses, but a good training in quality control, in analytical science, in biosafety, manufacturing, and IP and regulatory could be very useful for many of them ultimately to work on industry project or come in the industry. And then skills like what it takes to do scale up. How do you do your fund management and work on return of investment? They're very, very important. So before I start and start a project, I see that whether I have freedom to operate, whether I have all the IP freedom, okay, and whether it has commercial feasibility, so that I can get my money back what I'm investing. So those kind of basics should be there when you start a project. And working together as an example, for all the policy making bodies. I mean, we already are involved in different industry bodies, but I think here all academics and industry should come together and make sure that, especially for life science, And for the oceanography, ocean science, we need to have those policies friendly to support the science to move forward. In continuous engagement for academics and industrial research council and advisory board, many of the institutes already started. I mean, IIT Guwahati is a very good model. I work with them. CFTRI Mysore is a very good model. I also work with them. And so it shouldn't be just a project, whether there is a project available. It should be a long-term engagement. And one of the things I proposed in CII, a few months back is in academic institute, if you can create a position like industry affair position, you probably have a similar position who works with the industry. But if you have a dedicated role for industry affairs, they can connect and facilitate many of those things. And finally, I'll end with that to tell you that you are especially in India, you guys are in academics doing some great research, basic research, and as an industry, we have to take it to the next stage. So you bring it to certain stage, not just proof of concept, bring it to maybe TRL 3-4 level, and then industry will scale it up, work on the techno-economic model, late stage product development, business development, and finally generate the revenue which could be shared between academics and industry. So with that, I think my 10 minutes are over. I'll thank all of you for your time. My email ID was on the first slide. So if you have noted, please shoot all your questions to me. I'll be happy to answer. Thank you very much.
Thank you, sir, for elaborating on the various products from microalgae. We have a question from the audience. What are the regulatory requirements and tests needed to successfully approve the bioactives MNPs?
Yeah, bioactive MNPs, I don't work, so I'm afraid that I would not comment. I would comment on, like, if you ask about food, if you ask about feed, if you ask about materials like for food, we are working with the regulators, we are working with FSSAI. So bioactive MNPs, I don't work, and I'm afraid I will not comment.
Okay, thank you, sir. I have one more question. If NIO wishes to collaborate with you, what are your expectations?
No, I mean, so it's very simple. Is like if there is something, okay, like there is a particular project on microalgae, for example, which fits our requirement, and the expectation is, as I told, that it shouldn't be just like a proof of concept in the lab. If you have done up to a stage, okay, if you know that if you have an FTO, if you know that you're clear from your IP, and you have done to TRL 3 and TRL 4, let's discuss.
Okay, thank you, sir. Any more questions? Thank you very much.
Thank you. Thanks a lot. Let me see how do I share.
Yeah, a warm welcome to Dr. Sridhar Narayan, Director, Foundation of Neglected Disease Research, who will enlighten us on marine-derived therapeutic molecules. Dr. Supriya, Principal Scientist NIO, will introduce the speaker.
It is my privilege to introduce Dr. Sridhar Narayanan. He is the founder, director, and CEO of Foundation for Neglected Diseases Research, that is FNDR in Bangalore. Dr. Sridhar holds a PhD in pharmacology from Ohio State University and postdoc in neuropharmacology at the University of California. He has more than 20 years of experience in drug discovery and development. At FNDR, he has been responsible for the design and implementation of the scientific and business strategy for drug discovery and development. He is also the co-founder of Peptris Technology Private Limited. In addition, Dr. Sridhar is a mentor to several startups, academy and industry collaborations, etc. He has many publications and 15 patents to his credit. He has seen the discovery and development of one drug and 18 clinical candidates in different therapeutic areas. And he has also executed out-licensing deals with major pharma and in-licensing of candidates. This has generated revenues in excess of 180 million USD. Now I request Dr. Sridhar to please take over. Dr. Sridhar.
Thank you, Supriya, for the kind introduction. Are you able to see my slides?
Yes, yes.
Wonderful. Very good morning to all of you, and my sincere thanks to Dr. Narsingh Thakur for inviting me to be part of this wonderful session. It's such a pleasure to know about the work that is being done in the marine natural product space. So the topic that I'm going to talk about today is a collaboration between NIO and my organization FNDR. Particularly, this collaboration was between Dr. Narsingh Thakur and my group here in Bangalore. So just to give an introduction on my organization to all the members who are present here. So we are a not-for-profit company based out of Bangalore. We do extensive preclinical research in the infectious disease space. We have biosafety level 3 facilities for both in vitro and in vivo experimentation, BSL-2 facilities, cell culture microbiology facilities, biochemistry, molecular biology, DMPK, and even a small chemistry synthesis lab. So this allows us to do end-to-end preclinical drug discovery within our organization. We are approved by all the government bodies where the certification or approvals are required. And in terms of instrumentation, we are fairly well equipped to conduct any of the preclinical research. We do two types of work. One is our own proprietary innovative research, where we have more than 30 plus ongoing projects, 13 new drugs in preclinical development, 1 drug in clinical development, 4 repurposed drugs in development, and we work on a variety of disease areas. In terms of the services, we also provide contract research service on any of the models that we have developed based on the BSL-3 and BSL-2 requirement. We work with bacterial, fungal, parasitic, and viral infections. And the reason why we are able to do so many different projects is because we work extremely well in collaboration. And I will try to demonstrate that with one example from NIO. This is our drug pipeline. I wouldn't go into too much of the detail, but the lead candidate in our pipeline is TBA 7371. This is a molecule which is a novel mechanism of action, a novel molecule for tuberculosis. This is partnered with the TB Alliance based out of New York and the Gates Medical Research Institute. This molecule currently is undergoing phase 2 clinical trials in South Africa. And we hope that by the end of the year, we will have the phase 2 results and that we should be able to start some work in India with this molecule for clinical trials. So Dr. Thakur asked me to talk about a little bit about the different chemical classes of drugs from marine sources. I think Dr. Tapas Kundu gave a fantastic overview of this. So primarily, you have peptides, alkaloids, a lot of the steroids, polysaccharides, macrocycles, which are seen in the marine natural products. And these have been approved for a variety of indications, like Dr. Kundu mentioned, all the way from 1969 to 2018, you have a variety of drugs for different indications that have been either approved by the FDA or the EMEA or even by the Australian agencies. I'm going to now talk about PM181104. This is a molecule that was discovered by Dr. Narsingh Thakur in collaboration with the Piramal Life Science Group. And this was way back, I think, in sometime in the 2015 timeframe. So this came from a marine sponge. It's an actinobacterium of the Cocuria species, which has been broth extracted, and this novel molecule, which is a cyclic peptide, has been isolated from that. The yield of this molecule at the early stages was small. It was about 40 milligrams. Now at the preclinical level, we are able to scale it up to 200 milligrams per liter. And this molecule is active against a broad spectrum of gram-positive organisms. So all the chemical isolation, purification, characterization was done by Dr. Thakur's group at NIO. And this is just a structure of the peptide with a very high degree of purity of 99.2%, which was confirmed by HPLC analysis. And we've confirmed that the different batches that were produced by Dr. Thakur are also equally effective. So we've screened against two organisms, Staph aureus and Enterococcus faecalis, as our primary screening organisms. And against both of these organisms, the different batches have retained their potency. Also, in terms of the cytotoxicity, we have looked at two different cell lines, the THP1 macrophage cell line and the FG2 cell line. And against both of these cell lines, there is absolutely no cytotoxicity for this molecule. So what we have concluded is that this is a biologically active, reproducibly scaled, non-cytotoxic compound that we have isolated. So this is data from almost 110 Staph aureus isolates, which we have done at FNDR. And the bottom panel here shows the different strains, where MDR means multi-drug resistant, SDR is single drug resistant, and sensitive are sensitive to all drugs. So we have about 82 different strains that were sensitive, and against all of them, the molecule seems to hold on to its potency. Against 5 SDR strains that we tested, we had a very good potency, which was less than 1 microgram per ml. Against 17 MDR strains of Staph aureus, we had less than 1 microgram per ml potency again. In 5 of those, the potency was greater than 16 micrograms per ml, which means that the drug was not very effective against those 5 strains. So in terms of the overall percentage, it's almost working on 95% of the Staph aureus strains that are seen in the hospital. We've also looked at a few other strains of relevance, which is the Enterococcus family of organisms. And against all of these, the MIC and the MBC are very, very good and comparable to moxifloxacin, vancomycin. It's much better than methicillin, erythromycin, and linezolid and streptomycin. So as a new molecule which works on a novel mechanism of action, which seems to be on protein synthesis, we have a molecule which seems to work on not only sensitive strains, but also drug-resistant strains of the organism. We've also looked at Clostridium difficile, which is an organism which is of great relevance today. It is classified in the WHO pathogen list as a critical organism. And against this organism, the compound is super potent, right? So it is less than 0.015 micrograms per ml, and it's much better than any of the other gram-positive organisms. It also works on bacillary organisms. So we've tried Bacillus subtilis, and here the potency is less than 0.125 micrograms per ml. The major disadvantage of this molecule, being a peptide, is that it gets cleaved very fast on administration into the animals. And though we got extremely high Cmax levels with this compound, the half-life was very, very poor. And it was less than 20 minutes of half-life of this molecule. In spite of this half-life, we have tested this in animal models of septicemia. And this is a dose response for this molecule done in this model. And you can see very clearly that at 3 and 30 milligrams per kilogram, there is a significant reduction in the log CFU or the colony forming units in the animal model, suggesting that this molecule, if given frequently enough to maintain the Cmax and the AUC, will be extremely effective against the Staph aureus MRSA. That's the data that we have today. It's been a wonderful collaboration with Dr. Thakur, and we've had great success in moving the project forward. So, you know, as a motto of the organization, we always are open to partnerships. This is my email ID, and if anybody wants to contact me for any assistance with any of the drug discovery or development, my organization is very happy to partner. Thank you very much for your kind attention, and happy to take any questions.
Thank you, sir, for your talk. It would be nice to know in which therapeutic areas are marine-derived molecules useful and what is its present need?
So definitely, you know, oncology is probably the main area in which it has been investigated, but it can be useful in a variety of other areas, including infectious diseases. You know, there has been a lot of work during the pandemic on looking at antiviral compounds from marine sources, and there are definitely a few leads in that direction. So I would say that it's as such a highly unexplored arena when it comes to a majority of the disease states which are present in India. And I think there is a significant potential to explore marine natural products in a variety of these indications.
Thank you, sir. Any more questions from the audience? Thank you. I now welcome Dr. Raghu Kumar, Managing Director, Mycotech Private Limited, who will be speaking on omega-3 polyunsaturated fatty acids from marine microorganisms. Dr. Smita, Principal Scientist, NIO, will introduce the speaker.
Dr. Smita, we cannot hear you. You are not audible. You are not audible.
Hello, sir. Am I audible now?
Yes.
Thank you. Good afternoon to all. I'm glad to introduce Dr. Raghu Kumar. Dr. Sheshgiri Raghu Kumar is the Chief Scientific Officer of Avisa Myco Incorporation, a microbial biotechnology company. He completed his PhD at the University of Madras in 1973 on the topic of marine fungi. He later worked as a scientist at the Institute for Marine Research, Bremerhaven, Germany, for 5 years. He joined the CSIR NIO Goa in 1982, where he worked in the Biological Oceanography Division. His major interests have been the ecology and biotechnology of marine fungi. He is an internationally acknowledged expert of throstocritids. He was the project leader of the Marine Biodiversity Program of NIO and a member of the board of the International Marine Biotechnology Association for several years. He has published nearly 100 papers in reputed international journals and has a number of patents. He is also a member of the Research Council of NIO. With this brief introduction, I would like to request Dr. Raghu Kumar to deliver his talk.
Good afternoon. I'm having problems in sharing. I don't know how to do that. So let me, I'm afraid I'm not able to figure it out, but so this is, I don't know if this is visible to you. Is it visible?
No, sir, it's not visible.
Oh, so I'm having a problem in that. I'm using an Apple Mac, and when I click on the share, I get open system preferences, and it leads me to some screen. with let me check. I'm afraid if that is so.
So can you email us the presentation?
I
We are
could.
sending you the email address.
Yeah, I could. Yeah, thank you.
No. You ask on Gmail. It takes time.
Okay. You'll send it to me on Gmail, right?
Yeah, please send on Gmail.
Okay.
Rahul Kaushik C. Okay.
Just a moment. Yeah, please tell me what is the name address. Rahul.
It is there on chat box. Rahul Kaushik. Kaushik start from C.
Rahul Kaushik, all one word, right?
RAHUL CAUSHIK.
Yeah, at gmail.com.
Yes. First is
Yeah, come in. I'm sorry about that. Apple Mac has a Safari. Maybe I should have gone to Google Chrome and done that. Safari is often not compatible.
Dr. Raghu Kumar, have you sent it by mail, the presentation? Have you mailed the
Yeah,
presentation?
I'm just, it's just still going. It's almost there. Okay, it's gone. Okay, yep, should be there. I hope you got it.
Yes.
Yeah, okay. Okay. So I don't know if I will be able to see it on my screen, but nevertheless, okay.
Shoot.
So please let me know when it is on. Meanwhile, let me thank the organizers for giving me this opportunity. It's always nice to speak to scientists from NIO. I've been a lot of my life in NIO before moving over to the corporate sector. So I sort of know both the sides. And I chose this topic because I thought this is of relevance for both industries as well as academia. So there's a reason I chose it. There's a lot of scope in this topic for both to work together and even independently. So yeah, are the slides on, Judith?
He's just getting it on the screen, sir.
Yeah.
Yeah, but he...
So I'll try my best to keep it to 10 minutes less. Can't promise, but okay. So
are the slides visible to the audience?
No, not to me.
It's visible here.
Yes, it is visible.
Thank you, sir.
So
Thank you, sir.
it has not visible to me, but I'll go ahead.
So Dr. Raghu Kumar, you may go ahead.
Yeah, I have the PowerPoint with me here, so I'll go ahead. Slide. Avisa Micro actually is not working much on this aspect, but we are on to something else. But we are still interested in this topic, and we are doing peripheral work on this. But I did a lot of work on this topic when I was in NIO, and we even developed a technology and transferred to a biotech company sometime in 2000, 2002. So second slide. So this this talk is about polyunsaturated fatty acids or PUFAs, particularly, or omega-3 PUFAs. Particularly on top, you see the eicosapentaenoic acid or EPA, which is 20 carbon polyunsaturated fatty acid. And then you have the DHA or docosahexaenoic acid, which is a 22 carbon polyunsaturated fatty acids. And the third slide, please. I think I might have switched the second and third. This is a slide on the health benefits of the omega-3. In recent years, there has been a lot of interest on the health effects of omega-3 PFAS. You see a lot of advertisements and it's sort of proven that it's very crucial for the health of the heart and for the development of brain in infants and retinal sight. So there is a lot of importance attached to omega-3 PFAS, particularly DHA and EPA, of which I talked. Next slide, please. Okay. The C is a commercial source of omega-3 PFAS. Are we there? Are we there? Hello?
Yeah, this is on krill oil.
Yeah. No, not krill oil. Before that, wait, just hold on. Okay, yeah, krill oil. The C is the commercial source of omega-3 PFAS. Most of the commercially available omega-3 oil comes from fatty fish. The coast of Peru. So these are the major source of fish oil in the world market. Then come the krill oil, krill harvested from the subantarctic waters. So krill oil is another major source. And in this talk, I'm going to focus more on this microbial oil. These oils are derived from a heterotrophic marine alga, or I would call them fungus, called Orantiochytrium and Schizochytrium. The bottom picture that you see are cells are packed with oil droplets. So these two genera are the commercial source of microbial oil. So, okay, this is krill oil. Unlike fish oil, which contains mostly triacyl glycerols or triglycerides, krill oil is made of phospholipids. So it is more bioavailable, and there is a lot of demand for phospholipids derived from krill oil. That does not mean the fish oil market is small, yet it is krill oil is very popular. This is a global omega-3 market size. This is slide number 6, Judith.
Yes, sir.
in 2022, for example, if it is about $5.8 billion, in another 4 years, it is expected to rise much higher to 9.4. So there is a tremendous market demand for global demand for omega-3 oils. So there is a tremendous market, tremendous scope for both the corporates and, as I said, academia. I'll talk. So all this oil now, most of it, if you see the orange bar is for 2025 and the yellow bar for 2014, most of it still goes into functional foods, health drinks, health food, and next come the pharmaceuticals. Infant formulas are a very big thing for DHA because, as I said, DHA and EPA are crucial for the health of brain and retinal site. Then comes the pet and animal feed. The next one, please. Omega-3 market is now, there is a high demand for DHA. Now, as I said, of the two oils, fish oil and microbial oil, nearly 60% of the world market, or slightly more than that, comes from fish oil. Microbial oil from this Aurantiochytrium and Schizochytrium is growing, and presently it is at about 25% of the total omega-3 production. So compared to fish oil, it is a huge market also for the microbial PUFAs. DSM is a major producer, but there are also a number of small companies which are now making microbial oils. Next slide, please. The microbial oil that we are talking about, as I said, comes from this Aurantiochytrium, Schizochytrium. Believe me, you can produce, DSM claims that it produces 300 gram dry weight biomass per liter. Okay. So out of this, half, at least 50%, that is 150 grams, is in the form of oil. And of that, at least half of it is in the form of DHA. So you end up getting about 75 gram DHA per liter. Okay, this is what DSM claims. But we ourselves can approach this, not 300, but even 150 to 200 is good enough. And we have achieved that. But as I said, this is not our main focus right now. So these organisms are extremely good organisms for producing lipids. To my knowledge, there is nothing else compares to the amount of oil that these two genera produce. Now, what are the opportunities for both corporates and academia? Every strain of Aurantiochytrium, Schizochytrium has different levels of DHA. We have had strains with about 40% DHA in total oil to 20%. It can also have varying amounts of arachidonic acid, which is an omega-6 oil, which also is crucial for brain health. So, and these two genera are obtained exclusively from mangroves. And where else in the world can you find better mangroves than India? Okay. Unfortunately, there is very little work gone on in India to isolate different strains of this. There's a tremendous scope to isolate any number of variants with any number of variants in PUFA profiles from Aurantiochytrium. Next one, please. So in addition to the standard fermentation process, there is a lot of opportunities in molecular biology. I have not put the fatty acid synthesis pathway here. These Aurantiochytrium and Schizochytrium have a unique PKS anaerobic synthesis pathway, and the whole genome sequence are known. The whole pathway is known, and there's a lot of interest among molecular biologists to tweak it in such a way that you can either increase the DHA content or you can alter the fatty acid profiles by knocking out certain genes or overexpressing certain genes. For example, in this slide that you see, Liang et al., what they have done is these PUFAs that are in the middle can also be catabolized by oxidation. They can be used as energy source. So if you block that, they have knocked out the gene, and so the DHA contents have increased. This is just one example, and you will find some 50, 60 papers on similar aspects in literature. I'm not going into those details. Yet more opportunity for academia, I would say, rather than even corporates, is the looking at skeletonema. These are diatoms, and NMO has been working on diatoms for any number of years, 60, 70 years now. Skeletonema menzelii and Pheodactylum tricornutum have the potential for commercial production of EPA and DHA. Okay, and there are a number of papers which show the enormous promise that exists in this. So this is a process where you don't grow it heterotrophically in fermenters, but photosynthetically in raceways or in fermenters of growing photosynthetic organisms. So I'm coming to the last two slides. As I said, I would like to give you my views on what academia could be doing with the companies. In the previous speaker spoke about how the corporates approach any subject. What we do is we identify commercially viable technologies. Okay, the technology has to be viable in terms of market, in terms of IP or whatever. And then we develop them to completion based on, if it's a big company like Reliance, the previous speaker said, two speakers before, they have their own R&D team. They can do most of it themselves. But for others like us, we do a lot of R&D plus collaboration with institutions. So when we go to institutions, we look for specific strengths in the institution. We will go to them for service. We go to them at the last case, as you see, if the institutions have very unique technologies which are commercial up to that level, we may license it to them from them or negotiate various terms. And my suggestion for research institutions would be this. If you are into biotechnology, you need to understand how corporates think. So, which means you need to have a very strong tradition of science and technology. I will say you develop core strength in specific areas, whatever specific areas you are good at, and we will come to you seeking your help in areas where we lack expertise. And very, very often people ignore basic research because I believe that is the foundation for biotechnology research. If you have got novel frontiers, now there's a lot of interest in deep sea microbiology, deep sea microorganisms, proteomics, genomics. So from unique ecosystems, this is one thing. Finally, I will advise also that academic institutions should have a method by which they identify leads established by predecessors and build on those. I would personally say, for example, we did all this work on omega-3 PUFAS from Aurantiochytrium and transsotia technology, but that became the end of it after I left. Okay, there is still a lot of scope to carry on such research, and there's a lot of market, a lot of demand, a lot of expertise required to take this technology, to improve this technology. So to identify such leads, it should be it should also both be the responsibility of the institution to identify such leads that were established by predecessors, and also every individual scientist should have an entrepreneurial spirit of seeing where there is a possibility of the existing, even the predecessors, and how they can cash in on that. So that's it. I will conclude with thank you very much.
Thank you, sir, for dwelling on omega-3 oil, especially microbial oils, and increasing the energy source of PUFA. Any questions to Dr. Raghu Kumar? Sir, can you elaborate on the potential for marine-derived pigments?
Well, there's a lot of interest in marine-derived pigments from marine algae, right? I'm sure there are others who will know that much better. As Dunaliella, for example, or Hematococcus astaxanthin, or Dunaliella beta-carotene, these are all very well established topics. There is a lot of interest in other organisms which produce pigments like those, isoprenoid pigments, carotenoid pigments. And to my experience, this organism, there's a lot of interest in these Traustochytrids themselves, Aurantiochytrium and Schizochytrium, because many of them produce astaxanthin. So there's a lot of interest in trying to overexpress the astaxanthin genes and see if simultaneously you can get astaxanthin plus PFAS. There is a lot of interest in that. Yeah.
Thank you, sir.
Thank you.
So I take this opportunity to thank all the speakers of the plenary session for your suggestions and elaborating on important aspects. We will now proceed with the panel discussion on marine natural products, present challenges, and way forward. A warm welcome to our distinguished panelist, Dr. Vijay Kumar, Scientist G, Ministry of Earth Sciences, Commander Navnik Kaushik, Scientist F, Department of Science and Technology, Technology Development Board, Dr. Dheeraj Kumar, Senior Manager, Technical, Biotechnology Industry Research Assistant Council, DBT BIRAC, Dr. Taslim Arif Saeed, CEO and Director, Center for Cellular and Molecular Platforms, Dr. Girish Mahajan, Senior Vice President, High Media Laboratories Private Limited, Shri Tapan Chattopadhyay, former Deputy Director, Indian Chamber of Commerce, Shri Hemant Kulkarni, Senior Principal Scientist, CSIR, Technology Management Directorate, Dr. V.V. Sanil Kumar, Chief Scientist and Head, Business Development Group, CSIR NIO, and Dr. Bele Damodar Shemnoy, Principal Scientist, CSIR NIO. A warm welcome, and we hope to hear from you all about the opportunities in your area and what are your expectations from academia for fruitful collaborations. So as it's well known that the current pandemic spurred researchers to screen available compound libraries for properties that were not previously commercially interesting. Now we can imagine a crisis averted by building a diverse and robust toolkit of natural products. So the topic for this discussion is marine natural products, present challenges and way forward. And I would begin the discussion with our eminent panelist, Dr. Vijay Kumar, who's the advisor at MOES. Sir, all activities need funds, and if we have to venture further, so we request you to enlighten us on the funding opportunities for the industry and what are the type of initiatives that are preferred for funding.
Thank you. Thank you, madam. Good afternoon, everybody. First of all, I thank NIO for inviting me to be a member of the panelist on this thing. I think this is very important topic which are being discussed today, and I have listened to all the presentations. As such, my professional qualification is not in this field, but whatever knowledge I have got working in the Ministry of Earth Sciences related to this field, so I have tried to listen to the things. So in Ministry of Earth Sciences, recently we have launched a deep ocean mission. So it is almost, you can say, 4,000 crore project, and for a period of 5 years. And in this mission, we have 6 components. So one of the component is related to development of technologies for deep sea mining, underwater vehicles, and underwater robotics. Then other vertical is related to development of ocean climate change advisory services. It is implemented by MOES Institute in Quais at Hyderabad. Then we have another vertical, technological innovation for exploration and conservation of deep sea biodiversity. So this is, you can say, directly linked to the today's discussion. And then next is deep ocean survey and exploration. That is also little, you can say, indirectly linked to the today's topic. Then next vertical is energy and freshwater from ocean. And then last one is advanced marine station for ocean biology. So that is also linked to this topic. So out of these, you can say 6 verticals, 3 verticals are directly or indirectly linked to the today's topic. And under this technological innovation for exploration and conservation of deep sea biodiversity, so as such, it will be this thing will be led by a CMLRE, Central Marine Living Resources and Ecology Institute of MOES. And from MOES, NIOT is also involved, and then Bhabha Atomic Research Center is also involved. And then, of course, the other ministries, other institutes are also involved in this activity. So the basic objective of this particular vertical is, you can say, inventorization, archival, and of specimen and development of a DNA bank of deep sea fauna of North Indian Ocean through systematic sampling using remotely operated vehicles. So that is the main objective of this vertical. And in this, what CMLR is planning to do is to select a few sea mounts in the deep ocean, and then documenting the biodiversity of these sea mounts. And then they are also planning to do ex situ germplasm referral center for bioprospecting. And NIOT will develop the high pressure microbial sampling and culture system isolation and culture of deep sea microbes. And then they will also work on identification of novel biomolecules for culture based approaches. And basically the Bhabha Atomic Research Center will work on the experimentation on the process of origin of life friendly molecules. So these are the, you can say, more main components under this deep ocean mission. And more details can be given if someone is interested, like what are the activities, how that can be. And if some industry wants some participation or some other institutes, they can approach either CMLRE or they can write to us also, and we will connect them to the CMLRE scientists who are dealing with this thing. And there is a opportunity for, you can say, funding under this DOM also. Similarly, we are also planning for a, you can say, a advanced marine station for ocean biology. So as such, now the proposal is to set up this center in Goa, and then we will have the latest state-of-the-art equipment in this station. And anybody who is interested to do some studies, research, they can also approach and do that thing. But it will take some time to establish this center. So this is the basically main, you can say, activities which are being implemented under deep ocean mission. But as many of the activities related to marine biodiversity and all that is being done by CMLRE, which is the institute in under MOES. So thank you. I will end with this thing, and if something specific question or something is there, I will try to answer.
Yeah, thank you, sir, for elaborating on the various opportunities and programs going on. And also, it's nice to know that, industry is welcome and you all will be supporting them as and when possible. I will now go to Commander Navnik Kaushik. Sir, since you are associated with DST, Technology Development Board, what are the schemes available to promote industry, academia, collaboration, or corporations? Commander Navneek? Am I audible to you? Okay, I will move on to Dr. Dheeraj Kumar. Good afternoon, Dr. Dheeraj.
Very good afternoon.
Yeah, Dr. Dheeraj Kumar, he's a senior manager and technical in DBT BIRAC. So with your vast experience, of industry research and science management, how would you identify a compound or a technology to be commercially viable? And also, how do you attempt at, strategizing this translational activities from research to product development?
Okay. Uh, First of all, thank you all of you to giving me an opportunity to talk with you, all of you. Basically, I'm not from the background of aquaculture, but I was working on the field of parasitology, and earlier to Bharat, I was in US FDA. So yes, I'm from the healthcare background only, but I'm taking care of all the projects related with the aquaculture and biotech. So, what you are asking that how we are prioritizing the um, compound which is means commercially viable or not, isn't it?
Right.
So, in the if you are talking about the marine natural product, um, like we are, marine natural product which is commercially viable and to whom we are going to fund that only it will go to the industry level. So in this case, at Barrack, mainly we are taking that the compound which is means not at the early stage that now the academia company is just analyzing their some compounder library and then means heat analysis and everything means we are looking that the whatever you are proposing that it will be good for any drug or so first company or academia should prove at their in at laboratory level that it is working, and at least they should be at TRL 3 level, and then only we will then if they are coming to the barrack, then okay, we have already proved that it is working and it has some lead, and means we are not starting from the scratch, but we have already proved at level at the laboratory level, and it is working. So we are completed the initial study and we are reached at TL3, and now we want to explore and further study and prove it that okay or what were the our principle and it will work and it will go to the industry level. So first they will we want to do that a small animal study, and then for preclinical and clinical trial. So then that kind of compound Barrick is happily ready to assist them, and we are providing fund, and okay, then this compound will work, and we will provide all kind of support to them.
Yeah, thank you. So is there a special task force for marine biotechnology to support industry activities or a scheme to promote industry?
See, if you are taking in the at DBT we have task force, and from the means at DBT task force has supported more than 200 projects in the field of this aquaculture and marine biotechnology. But that is not means, but all the 200 projects are not that means academy or industry collaboration, but that is from the industry, maximum from the maximum from the academia. But at Barrack means DBT started Barrack only for this promoting industrial private investment, and through our CBRIA scheme and BIPPA scheme, we are promoting different startup and industry to work in the field of this marine biotechnology also. Means, in fact, we are promoting every kind of activity, but if you are asking for the marine biotechnology, so marine biotechnology aquaculture is also a part of our this investment funding scheme under this BIPP and SIBRI, and this BIPP and SIBRI is mainly for industry. But we have also another scheme, that is PACE, in which academy, we are promoting academia and industry partnership. We have a project at NIO also. We have funded a project with to Dr. Thakur for developing this in collaboration with FNDR. I think it is for anti-parasitic drug only, I think.
Antibiotics.
Yeah,
Antibiotic.
antibiotic, and it is in collaboration with FNDR. So means we are promoting academia industry collaboration also, and we have in BIPP SIBRI we have also funded several company, and we have already few products are in the market, from the Bangalore based company Aristogen, who is selling that Vibrosild to the anti-vibriophage probiotic developed by Aristogen, and which is double standard RNA specific to genes of WSSB to control WSSB infection. And also we have funded this C. Abdul Hakim College, and in collaboration with this GPS Biotech, and also that Thibana Meditative and Nutraceutics Private Limited from Chennai. They are selling that DS-stable nano-sized formulation of beta-glucan particle. And this Aristogen has also developed this EcoSense WSSB detection kit, a single tube nested PCR kit. So we have this means funded many industry and academia collaborative project also, and also there is a scheme in collaboration with means DVT Barrack Joint Call, that is ATGCA scheme. In that ATGCA scheme, we are also promoting this academia industry collaboration, and in the both means PACE, there is a in PACE, if I are talking about the PACE scheme of Barrack, so in that there are two components, AIR and CRS. So in AIR we are promoting academia to work on the field of means to start with a scratch with means limited idea, and to go to the TRL3, means in AIR they can work and reach to the TRL3, and from TRL3 they can apply under the CRS of the PACE, and from that we are means they can reach to with come with the industry collaboration, and prove their idea that okay, we are now TL3 and now we wanted to go to the TL7 or means preclinical trial and everything. So under that we are promoting both industry and academia from means with the in the pace we are promoting from TRL2 to TRL7, and in fact commercialization step also. And in similar in the case of ATGC scheme, the starting level of the project should be at TRL4. So both this scheme, this ATGC DVT Barrack Joint Call scheme and this PACE Barrack scheme, it is for academia industry collaboration.
Yeah, thank you, Dr. Dheeraj. It's nice that you all are supporting industry and academia. We have Shri Kulkarni who will add to some comments.
Am I audible to everyone?
Yes.
Thank you very much. I think since morning we have touched upon diverse areas which are of commercial interest. As I look at it, each of the scientific departments, funding agencies, they have their norms. Some are funding at R&D, some are funding at the pilot level, some are funding at the commercial level. But as we look at the macro level, especially a 2030 position, I seriously believe that our models need to change. These need to change in accordance to the requirement of the industry, in accordance to the dynamics, markets, and the investments. And we are talking about the huge kind of investments. Reliance is putting in a lot of money. They are making their own investments. Other mega corporates also are making, but still, India as such is not positioning itself. How do we bridge the gap is a key issue. In the morning, one of the suggestions that came up is that let's have a brainstorm with key select holders, key companies, to identify areas wherein collectively we can look at the S&T interventions, policy interventions, other regulatory interventions, along with the fiscal incentives. Fiscal incentives are important because it's a huge investment from the corporate side of also, and it's also important to meet the SDG requirements from the India's point of view. So I think it's a fair idea to look at the whole picture in a holistic way and develop a funding or a strategy to reach to those goals. So my take with regard to the funding opportunities or to the way we should move forward is to look at this particular aspect of marine living resources and how do we capitalize or monetize the S&T, the expertise that we have through industry participation requires larger deliberations. And obviously, we need to sit together, the TDB, the BIRAC, the DBT, CSIR, and whichever the funding agencies, along with the industry components. I think there's a lot of ground that we need to actually cover. And many of these things, especially the last mile connectivity, the marketing, we haven't really discussed. We are all saying that there is a need, we need to develop the technology, import substitution, but we haven't really drawn a path and piloted at least one or two to test the waters. So, I mean, my submission would be that as a takeaway from this meeting, we should also look at creating a macro picture, maybe a white paper or a roadmap, and take one or two areas which are of great importance to the industries, where I should say low-hanging fruit, and try them out through a proper structuring and proper financing. Now, if we need to tweak out certain guidelines for our financing, we should do that in consultation, obviously, with the Ministry of Finance, Niti Aayog, and I'm sure they will support it. That's all from my side, but I would like to have your reaction to this. Thank you.
Dr. Dheeraj?
Yeah, sorry. Can you repeat what is the specific question?
Dheeraj, did you hear what I said, or?
Means yeah, I hear that you are asking about that all what this means market tested means and what we will do.
No, no.
Means, but
The broader picture, Dheeraj, is today we are sitting on a very important eye-connect event. We are trying to reconnect with the industries so as to position S&T expertise that exists across the S&T fraternity in India, be it CSIR, DBT, ICMR, whoever it is, publicly funded S&T diaspora, I'm saying. And we need to understand two things. One, the relative advantage that each party brings to the table, whether it's an industry or whether it's a S&T fraternity. Second, what is the market complexity? Which are the products which can be taken forward, and in what way this consortium can jointly prepare a roadmap? As I was mentioning, there could be a fiscal incentive that is required, maybe policy, maybe some kind of a structuring. So there are n number of things that go hand in hand. So I think it's important for us to sit together, and iConnect event gives us a platform to start off that dialogue and perhaps identify 2 or 3 areas in this MLR, that's marine living resources, and say that if we take up these steps, I'm encompassing all the things together. Finance is not the only one, but I think holistic approach may be the need of the hour. And I'm sure technically CSIR, DBT, and other institutes are capable of reaching out to a level, and industries should also chip in to take that forward from that level to the level which is required for the commercialization. So this was my thought. I was just seeking out the reactions from all the panel members so that an action point or an outcome can be defined for this event as to how do we take this forward beyond this point. Thank you very much.
Means whatever I have read from the literature and everything, because I found that this marine natural product means there are several means pitfalls like the this material supply is also a drawback for drug development in marine natural products because they are obtained from the natural sources and usually in only very small amount. So means we can work on that area also, like chemical synthesis, that's a compound or semi synthesis, the means we can use that kind of technique, and we can promote academia and industry to work on that so that this supply should be increased. And also means
Thank you,
I found
Dheeraj. I
that
think we got your point. Can we move to Dr. Varke and the industry experts that we have here?
yeah.
Yeah, invite the panelists to comment.
Yes, sir. We agree with your concern that there should be more interaction between the industry, and we are more than happy to give our inputs. The CSIR has been very proactive with us so far, and DBT, if the BIRAC comes in and gives us some support, it definitely can boost this, because this ecosystem has to be developed. It's not just the industry, but we have to from the government, we need to set up an ecosystem with the fishermen where we can cultivate these seaweeds. And we've seen one project successfully happen down south in Tamil Nadu, but they're restricted only to one product. But the government will definitely need to pitch in for developing and encouraging the fishermen to, because this is a very low hanging fruit in terms of cultivating seaweeds. They don't have to do much. There's also a lot of women empowerment in this, because most of the time the women who don't go for fishing are there at home, and they can take care of these fields, and they can cultivate and manage these fields. That's what's happening in Tamil Nadu, it was observed. And the same thing can be done in other states like Gujarat, Andhra. I've heard the Arabian Sea below Maharashtra is a little difficult, but there are different ways of doing it and can be done. So yes, there is huge potential. We as an industry ourselves require, as I said, a few hundred tons of the seaweed, and it could be a big potential. And if BIRAC and the Ministry of Fisheries, Marine will rope in and give some kind of subsidies or some kind of encouragement training with the help of institutes like CMCRI, I think it can create a better environment for the future. And as you said, it needs to be on a multi prong. There needs to be a multi prong approach, right, from industry to the fishermen, and the market is there. So we feel the market is there. We saw since morning so many different products are coming out of the marine environment. But at the end of the day, who gets the product out of the marine environment? It is not the industry, it is not the institute, it is the fishermen or the people who are working at that level. So if that ecosystem falls in place, if there is uh, jobs for the people at that level, then all different products can be harvested from the oceans.
Yeah, thank you, Dr. Varki, for your comments. I will go to Dr. Taslim Arif. So, as you head the Discovery to Innovation Accelerator program, and you have the supporting translation of discoveries to application, entrepreneurship, and technology development, how are scientists or entrepreneurs encouraged to start up companies? And what are the opportunities for the startups related to marine biotechnology?
Thank you. Good afternoon to everyone. I'm Taslim Arif Syed from CCAM. But just, I think it's wonderful to be here on this platform and hear before that from Shantanu, Sridhar, and others who spoke very eloquently in terms of the kind of work which was happening and now part of this discussion. Thank you, Dr. Thakur, for the kind invite. Quickly to respond to, I think, coordinator's comment and a question. I think there are significant opportunities in terms of how science that gets generated in labs, be it in academia or industry, could be taken forward through the value chain or the process chain in terms of the readiness of the technology. And there are, for last 10 to 12 years, there have been significant, new schemes and funding opportunities which are being built. We all know that slowly and steadily, clearly, at the central level and possibly at the state level as well, there is a significant boost towards translation research, towards application research, and even possibly at the all funding agencies and their institutes, there have been a promoting and encouraging that kind of research. This gets backed up by funding, both from the main funding agencies like DBT, CSIR, DST, and others, as well as their arms which promote entrepreneurship and industry engagements like NSTDB or BIRAC, or many others, including Atal Innovation Mission and so on. Together, there have been very large amount of funds which are available, and every year many new such research gets supported either through the translation schemes that Dr. Dheeraj Kumar from BIRAC just mentioned. These are very large numbers which get actually funded when an academic scientist brings together the discovery that he or she has developed in his or her lab and projects a potential application down the line, and seek support for the validation cycle through the value chain of the technology development. And there are several schemes which are being built up. This is for an academic scientist straight on for the translation work. And there are several task force at different, both funding agencies which are supporting them. In addition, there are several entrepreneurship opportunities for scientists, again, from different, the walks of scientific fraternity, academia, industry, or even outside that in a not-for-profit research organizations, where these scientists are provided very good, handsome amount to validate their ideas at the early stage through a proof of concept study. Schemes like BIG, there are DST schemes and STDB schemes like Prayas and many others, which I'm not even able to pick up, plus funding from Atal Innovation Mission, and the state level like Karnataka has Elevate scheme, and I'm sure many other places like Goa, Maharashtra, and many other states have their schemes, where scientists are being provided funds for proof of concept studies with a larger ambition of building a technology or a product down the line. And this actually has come significant way. We have now a truly remarkable work happening. And if I may just bring in now the focus to the marine biotechnology work, I'll give you a very quick example, some of the work that is happening. Shanu already talked about the algae that the Reliance works on. We have a company called C6 Energy, which has been working on it for many years with its plants in Tuticorin and Indonesia, where they have been using algae for various aspects of impact, including A, biofuel, B, agricultural growth enhancer, and they have a collaboration with Mahindra and significant product in the market. And now they are building several other products for possibly other non-human feed applications, which is phenomenally attractive in going forward to support those aspects. Plus, there are several efforts Sridhar mentioned of the products for pharmaceutical work, so that is going on. And then there are several more efforts which actually we are seeing now in terms of what you would call it the combination of medicinal chemistry and the marine microorganisms based approach for development of new molecules, either nature inspired molecules as we call them, and so on. So there are several, I will possibly not go on that, but what I wanted to share with the entire fraternity here, which has come to this platform, there are very good opportunities. Marine biotechnology for me seems like an untapped completely opportunity, and collectively there are ways where we can come together and possibly have a dedicated effort and a consortium with a mission driven objectives of building new therapeutics, building new biologics, building new One Health products to ensure that this science that is being generated taken forward. And I'm sure we can do it all together. I'll stop here and happy to take questions. Thank you.
Thank you very much for your informative inputs to our audience. I will now go to Dr. Girish Mahajan. Good afternoon, sir.
Yes, good afternoon.
Yeah, sir, you have been associated with various renowned organizations such as Pyramid Enterprises, Unilever, Johnson & Johnson, including NIO, NCPR, and ICT. What are your experiences with, these collaborations with research institutions, especially NIO?
Okay, thank you first of all for inviting me on this discussion panel, and thanks to Dr. Thakur especially for opening this forum for me. And basically, I would like to share, like whenever I was with Pyramid and now in High Media, so we always realize that collaboration is the key of getting success towards new products. And if I go back to my career in Pyramid in 6 years ago, so we had a very long collaboration with NIO, and especially with Dr. Thakur and his earlier team at that time. And we have a lot of successful drugs. And I'm really glad to see that Dr. Sridhar has presented one of my molecule along with Dr. Thakur, that is PM181104. So it is actually a sponge derived compound, sponge associated bacteria, Purpurea. And we got the first hit at that time when we started the marine program with Dr. Thakur. And it really led to a good confidence that yes, marine is really a good source. One good advantage is that when industry and academia recognize the core expertise of each other, like why we started with NIO at that time. NIO has a good insight in collecting the deep marine samples, and getting oceans marine samples is easy, but deep marine and the sediment from the is a very expertise in NIO, and the team was good in bioorganic modifications and evaluation. So we got collaboration with NIO, and during that they used to supply us samples from various unexplored sources, especially the deep marine samples, and from that we got number of hits. And PM1104 is one of the success. We got many antifungal also from that, and some Salinospora species, a good number of anti-cancer. And along with NIO, we were also in touch with NCARO. Again, it is one of the marine but Antarctica related institute. And there also we got a very powerful anti-TB compound, PM181108. So of course, it means the marine resources are really untapped, and there is a huge scope to work on these marine resources. And as I mentioned, we have to understand the expertise of each other. Now, similarly, currently also we are in connected with CSM CRI. So we understood their expertise in cultivation of these seaweeds, and it is going bit at a lower pace, but due to Corona also it got hindered. But now again it is taking up the pace, because we have to understand sometimes the pace of each institute also, and the changes which happen different institutes. So that way, in one line, I can say this marine resources is really untapped, and we have to come out with new ideas to explore this. And at the same time, as mentioned by some of the earlier experts, that we have to make a sort of wish list, what industry needs and how this academic institute can pitch in to get the relevant products through the various funding agencies, as mentioned by other Dheeraj Kumar and Dr. Dheeraj Kumar and Dr. Tasleem just now. So I think it will be a good idea to move ahead with more such resources. Thank you.
Thank you, sir. It's nice to hear that you're very happy with collaborations with NIO. Can you share some tips to a successful collaboration or commercialization of these initiatives?
See, many times, especially in case of bioactives, I can say bioactive molecules, it's a long process, because before clinical studies also it involves a lot of studies. And the way like Dr. Sridhar also explained you, right, before it goes to clinical, we have to establish the complete in vitro and in vivo profile, and also the toxicity studies, safety studies, formulation development. So these are to be, means again, one particular organization may not be having this expertise. So we have to again have the CRO help and collaboration, or if particular institute has some experts, professors into that area. So we have to explore that opportunity. And especially in case of in other hydrocolloids, if you can say what I've experienced here, that initially it is they are most of the academics, they are excellent in the work at a lab level. So to take it ahead, it needs handholding from the industry. So that is what we are doing exactly, so that it is getting into pace. Now we can say at a pre-pilot scale. Now later, once the pilot scale is completed, especially in hydrocolloids, so we'll move to the commercial scale. So understanding core expertise and other experts outside the institute is very important to
Yeah, thank you, sir.
successful.
Thank you very much. I will go now to Shri Tapan. Good afternoon, sir.
Good afternoon.
Yeah, sir, since you have a rich experience, in the spectrum of industries like pharma, chemicals, technology, seaweed, and also in the Chamber of Commerce, have you also headed, and you have also headed the supply chain and operation. So in this respect, what would be the, products industry requires, and what are the challenges from industry point of view?
Thank you very much. Thank you, Dr. Thakur, for inviting me as a panelist into this program. I just want to touch upon the two segments. First of all, as ocean is very big, so ocean has also got a lot of products which cannot be said in this very small, short time. What I want to emphasize is that the industry, the application of the product that comes from the sea or ocean or seawater is huge. But I just want to emphasize two areas. One area is that it's the inorganic part. Now, till now, we have understood, we have listened so many products of the biotechnology or biochemistry. I just want to emphasize on the inorganic part of the products that are available from marine. If we take out the salt from the seawater, the mother liquor, which is basically called as a sea beetle, has got the potassium, magnesium, and bromine and other materials. Now, as far as the salt is concerned, 75% of the salt is manufactured from the seawater in India, which is around 20 million tons per year. But the rest of from the rest of the water, there are possibility of having the material. First of all, I will talk about the potash. As far as the potash is concerned, India is 100% dependent on imports. India doesn't produce potash at all. And India spends around a huge amount of money, 10,000, 11,000 crores for this particular segment. Now, there is a technology available in India. This other part is a magnesium. And this potash is used for the agriculture use all across the country, which is a huge quantity. Now, second product is the magnesium oxide, which is derived, these can be derived from the seawater. It can be used in various kinds of, refractories. It can be used for magnesium metal, which can be used for the batteries, automobiles industry, defense. If the question is that whether we are capable of producing, yes, the technology is available in India. And therefore, the import substitution can be possible. And once the import substitution is possible, this product will be manufactured in India, and it can cater to the refractories. It can go to steel. It can go to metals. It can go to automobiles. It can go to cements. So across the spectrum of the industry, this material is in scope. So in this case, the major challenge is that how this can be commercialized. I just want to mention that all the technologies that are available in the CSI laboratories, in CSMCRI, SICRI, in ceramic and glass technology in Jadavpur, and also in Jamshedpur. So therefore, the challenge of this particular segment is how to commercialize these technologies to be Atmanirbhar Bharat. The second portion is the seaweed. While working on seaweed for last 5-6 years and organizing several international conferences, physical interaction with these experts of India and overseas, where Dr. Girish Mahajan, then Dr. Rahul Varke was a part of my team, and we have all worked together, along with the C6 Energies, the Aqua Agri, and all other companies. The government of India has already earmarked 640 crores for the cultivation of seaweed. They have asked the state governments to implement, or there is some hindrance which needs to be cleared at this point of time. First is that the sea area has to be earmarked on the ownership backed by a national feasible policy. Because investors or entrepreneurs will not invest money on a particular water body. They must know that where they are putting their money. And then only they will come and set up the cultivation. Number two, that there is a requirement of processing the seaweed which can be procured from the sea into a product for the commercial applications. So there needs to be a development of the industry. And the government of India and the state government has to make a policy for suitable for the industry into their own area. Fortunately, Tamil Nadu government is going to have a seaweed park, which will be the integrated complex of a research development, manufacturing, marketing, and export. And third is that the Indian coast has got 200 species of seaweed. But in India, we have Cavaficus, one, and Gracilaria, which Dr. Rahul has informed that Gracilaria and Gladionia, these two can be cultivated. Sufficient areas have been identified by this time. I want to mention that there is a alva grade or species which is produced in the Sundarban area in West Bengal. So there is a huge scope of cultivation of the seaweed, provided that the scientific institution should earmark that which species will be cultivated in which area. Now, finally, what I want to say is that all the research that is being done in the CSI laboratory, let us have I personally believe that there will be more and more interaction between industry, research institution, and the government. The three divisions should come together and establish that whether a particular technology has a real viability in terms of its technicalities, or at the same time in terms of commerce, then only the India can really fulfill the dream of being Atmanirbhar. Thank you.
Thank you, sir. Director, sir, would you like to comment?
Yeah, just one query.
Yes.
Why India is not producing any potash? If you see our English era, when means say 1600, 1700, 1800, India was producing a lot of sodium nitrate, and that was the major source for England. So why it means we are not continuing that?
See, I tell you, there are two aspects. One is that the application of the potash in fertilizer is necessary for the development of the crop. But the absorption of the potash in the field is very, very low. But as we do not have a technology to manufacture potash, technology is available now, but the technology has not been really explored to get this into a production mode. So today, India is importing I mean, the other day I was listening that around 20,000 crores of the subsidies is being given for this potash import. Now, the actually the low grade potassium is required, which is not also available and is not being imported. So, to answer your question, the technology was not available so that the industries has not been industrialist has not been invested to that extent, and all through these days the potash is being imported. But that is why I'm telling that we have the institution which has got the technology to manufacture potash. So why not we'll take up, if we cannot substitute 100% of import, at least if we can substitute 30 to 40% of the import, there will be huge savings.
Thank you, sir. As you spoke about seaweeds, I would request Dr. Sanil Kumar to speak about, the initiatives at NIO, which are being taken with respect to seaweed research. Dr. Sanil Kumar?
So I thank all the panelists for the covering up all the different aspects of these marine natural products. So now, because we have already, Dr. Thakur has told that we are since 1990 onwards we are working on this marine natural products, and earlier also we had a good industry association like Shriya and all, and in 2000 and all, even we went up to clinical trials of up to 2, but unfortunately sometimes that acute toxicity and such things has happened. So because there will be a lot of screening and things like that. So after that, with the industries means like Thakur has already mentioned, and already FDNR and then Pyramel, all this we had a very good active collaboration, and presently also now we are having with the melanin with the green lay. So all these things already Thakur has mentioned. So apart from that, actually NI also do a lot of, because we are having a multidisciplinary means all the experts are available related to ocean science. So we are having core biologist and biotechnologist, then also geologist, physical oceanographers, chemical oceanographers. So identifying a location for a seaweed farm or something. So because then in the ocean, so we have to look for all the aspects. It is not only that biological thing, because it is it should allow when we do a farming. So that what is the physical conditions like? So some of the areas are very active area where the physical currents will be very high. So that whatever rope or whatever that we do, so it should survive in that condition. So all those survey, so which presently we are doing that. So we are identifying locations for the west coast, some locations. In it's again a multi-institutional project with the CSM, Sarai Bhavanagar and NIO, means like that many institutes are involved. So that is one thing which we'll be working on that.
Yeah. Thank you, Dr. Sanil, and thank you. I will continue with Dr. Sanil. So you have been heading the business development group at CSR NIO for several years, and also you've been leading a lot of industry-funded projects. From your over 30 years of experience here at NIO, can you give a brief on the efforts and strategies used to work with industry and other agencies?
The result just now I told, actually NIO is very strong in the basic research, because as Dr. Raghu Kumar, means earlier panelist has mentioned, that means to develop anything like means our foundation will be on the basic research. So that part, so we are working means from the beginning onwards, and as I told just now, the 1990 onwards, that marine natural products we worked. So then that resulted into identifying different molecules and so many biological products. So then but getting up to this product also is a very difficult task, because again because cost effectiveness, because earlier Professor Kundu also mentioned that, because some of the things which go in the deep ocean or somewhere, that getting the isolating the compound means for the deep water only we have to get. So like a facility, so facility wise anyways have a good facility. So apart from that, what happened is we are dealing with the industries for any ocean related problem. So whether it is a site selection for a farm or a what is the design parameter, like if you wanted to develop a case culture or something like that. So what will be the design parameters to be taken in that area? So this way we have done a lot of work doing surveys for all industries like chemical, pharmaceutical companies which are coming along the coast, because they use a lot of water either as a seawater intake or a effluent disposal. So identifying the location, laying the pipeline. So all this work we have done in the past. Now then apart from that, this related to marine natural products, that's what I told that some many collaborations with the industry thing, because our basic research will lead up to TRL3 only, so we are not progressing further. So anyway, all the panelists has emphasized, including Dr. Kulkarni from CSIR, that the industry the institute industry collaboration is a thing, but the only thing is from that what are the potential industrial applications and the materials needed for that particular activity. So identifying that is more important. Then is there a space for that bioactive in the, or is it an innovative thing? Then what is the formulation desired, and what will be the target price of the final bioactive? And we have good lab facilities, but again when you go for want to do for scale up or things like that, that is a place where we are looking for a industry collaborations to have a large scale production.
Thank you, Dr. Sanil, for briefing on this issue. We now have Dr. Bele Shenoy. Good afternoon, Dr. Shenoy.
Hi, good afternoon, ladies and gentlemen. Hi.
Yeah, Dr. Bailey is a fungal taxonomist. So, Dr. Shenoy, you have served academicians and industry on fungal identification and taxonomy issues. So, as a researcher and with your interactions with industry and academia, what are your views on the importance and the future prospects of marine biodiversity and biomolecule research?
Thank you, Dr. Judith, for asking me this question. It's a very important question. I'm a big fan of marine diversity studies, because marine life or marine biodiversity is the backbone of marine biotechnology. So we don't know many things about the life in marine systems. So the unknowns are almost infinite, and our oceans offer us limitless opportunities to explore and discover novel organisms. These could be novel in terms of taxonomy. These could be novel in terms of their functions. And this kind of research must be continued, and this kind of research must be conducted regularly, because there are so much of unknowns that have to be discovered, right? But marine biotechnology research is a different ballgame. So here we need to consider the requirements of the stakeholders. We need to consider the aspirations of the stakeholders. So what kind of research problems, as somebody who is getting into applied research, we are trying to answer or question ourselves. I mean, what kind of research questions we are trying to answer, that will determine the future course of marine biotechnology research. Thank you.
Yeah, so we also come across a lot of, basic research. You must have come across when you were doing your research. And so what could be done to transform it into, the applied research, or make it, attract industry?
Yeah, thank you for that question. Again, see, applied research exists only in presence of basic research. So applied and basic research both are required. But as I told you, in applied research, we should consider the requirements of the stakeholders and their aspirations, and we should actually constantly be in touch with our stakeholders, try to listen to them, and try to understand the requirements. This could be done through this kind of online meetings, offline meetings. We could use social media tools like LinkedIn, right? And when you start your applied research and interacting with your stakeholders, but finally, when you have the technology ready in your lab, you would know who to talk and who to transfer the technology to. And another important aspect that I want to emphasize here, as an academic researcher who is inclined towards applied research, it would be best to start the applied research with the patent landscape analysis. So with this kind of analysis will give will let you know the current status of a specific technology and what are opportunities ahead actually. Judith.
Thank you, Dr. Shenoy. As we were discussing during the panel discussion, I caught a point that some of the panelists, they spoke about deep sea microbes. So I would like Dr. Girish Mahajan, if he could elaborate on the potential of these deep sea microbes for industry. Dr. Mahajan?
Yeah, see, basically, since we had worked with NIO, and NIO periodically used to provide us the deep sea samples, and that is where we got about almost 1,500 different species of actinomycetes and eubacteria at that time. And as of I remember, we got at least 13 NCE, new chemical entities, from this deep sea. And there were a couple of Selenospora species as well, which is a gold mine for anti-cancer compounds. And from those 13 NCEs, almost 9 were anti-cancer and remaining were antibacterial. So obviously, it shows that this deep sea sediments have a tremendous potential. And among these, one of these PM1104 is among this one of those 13, which is going ahead with a lot of efforts from NIO and Dr. Sridhar Narayan. And also we have got similar samples from Antarctica. Means I don't know whether they are called sediment, but labels were as a sediment. So obviously there also we got a number of hits, and more than 3,000 cultures, actinos and eubacteria identified from there also. So overall, I can say deep sea sediment has a great potential to explore the novel bioactive scaffolds and to address the burning need of the new antibiotics and anti-cancers. And again, I will say that these marine microorganisms, though they are tested for a number of things, but the majority they have potential in anti-infectives and anti-cancers. Thank you.
Thank you, sir, for your comment. But since this comes under the Deep Ocean Mission to support the Blue Economy initiatives,
Right.
I would also like a comment from Dr. Vijay. Yes, sir. Sir, I would like you to comment on the possibility of supporting this type of, aspect or research.
Yeah, thank you. So I think the points raised by various panelists are very important. So there is a little, you can say, lack of interaction between the government, then the institutes, and then also the, you can say, private players. But I would like to say that under Deep Ocean Mission, because we are working on the 3 components which are directly or indirectly related to the biodiversity. And one of the component is related to polymetallic sulfide program. So under that also we are doing some studies related to the identification of polymetallic sulfide sites. And there whatever organisms are available, I think we have not studied that in detail, or I think there may not be any study at that type of water depth. So these, you can say, bands are available around 3000 meter depth. So we are also trying to collect samples at that depth also. And then there is a possibility that that samples, whatever we are collecting, it can be used by the, you can say, various institutes and the private industry also to do it, to analyze it further. So as such, like this work is done by NCPR Goa, and but they are only collecting these samples related to the contract with the International Seabed Authority for polymetallic sulfide program. But I think because India is putting up, you can say, it is a very costly affair of collecting the samples from that depth. it needs, you can say, big ships and then the collecting equipment and everything. So if the collected samples can also be used for some other, you can say, commercial purpose or analysis, I think that will be very useful. So if
Thank you.
somebody is interested in that, I think we will also be happy to collaborate.
Thank you, sir. Thank you very much. I wish to thank all our panelists for sharing their views, which I'm sure will be beneficial to our research community and industry, and it will also help to draw a roadmap in this initiative. I now request Dr. Anas, Principal Scientist, NIO, to give the closing remarks.
Good afternoon. Good afternoon, all of you. And now we have come to the conclusion of this ICONACT meeting on marine-derived products having potential industrial applications under the E3OW scheme of theme of CSIR. This has been a productive meeting and that good progress we have made. So thank you all for taking part and contributing to this successful meeting. Marine ecosystems comprise a diverse group of organisms and ecological processes that can be explored for developing products and processes for solving many of the problems faced by humans. And this may include, or this may be extended from waste treatments to minerals, food supplies, cosmetics, fuels, and novel drug molecules. CSR NAO has been conducting several research programs to understand the seas around us and translating the information for the benefit of all. The ongoing research program bioprospecting under the E3OW scheme focused on translating the science into process and products of human benefit, and interactions with industries are very important in this line. The meeting conducted today was organized with the aim of providing a platform for one-to-one interaction between scientists, technologists, industries, funding agencies, and policymakers in this area. We are very encouraged by the many concrete proposals, ideas, and strategies that have been discussed in the meeting to promote the industry institution collaborations to explore oceans for novel products and processes having industrial applications. We had many innovative ideas and proposals that include impact investment and private-public participation models for larger role of funding agencies. In the introduction, welcome speech by our director and the talk by our team director and the project leader, they have emphasized the importance of exploring marine living resources and importance of collaborative research in the field, strength of CSIR, NIO in oceanography research, and the translating the technologies that are already in the different stages of TRLs and the importance of industry academia collaborations to validate their economical fitness and convert them for a final use by the users. So CSR NIO have 2 research vessels, research laboratories, more than 25 researchers working in marine biotechnology, and have more than 5 research technologies, which is already at in the different stages of commercialization, and 7 leads at different TRLs levels. So when starting the program, a great introduction on bioprospecting of marine resources and drugs available different stages of phase trials and available in the market are included, given by Professor Kundu. And he emphasized the need of organized approaches for the development of antibiotics for addressing the antimicrobial resistance from marine sources and potentials of using marine-based food for achieving the sustainable development goal targets related to health and food security. Subsequent talks by experts from academia presented their facilities and research outputs, which are at different stages of TRLs and status of association with industries in the area. The technologies on probiotics and prebiotic combinations for waste management from linoleic acid. These are some of the examples from CRI. Experts from industries also emphasize the importance of industry academia interactions in the area of marine bioprospecting. Industries will be happy to associate with CSIR for developing technologies which is at stage of TRL 3 or above. Such collaborations are required for reducing our dependence on other countries for raw material for the production of many valuable products. An example cited in this line was hydrocolloids, which have a global market of 23.2 million. And these topics were again deliberated in detail in the panel discussions, and we made some concluding remarks which can be taken forward. And this includes when the marine resources are untapped, and the panelists and the experts met together, agreed the need for collaborations for harvesting these resources for developing products and process for human benefit. Institutions and with their research facilities can take up the research up to a maximum of TRL 5 or 6, from where to jump to the final level of TRL 9, we need industrial collaborations. That is very much required. So importance of identifying the key expertise of industry and academia is important for each project at this level. And then the encouraging research programs and entrepreneurship is important. Prioritizing the research needs in marine biotechnology and financial support for the marine biotechnology was discussed in detail in this panel. Different schemes are currently operational from Ministry of Health Science, CSIR, DBT, and DST for promoting research in the area. Deep sea microbes have been identified. Deep sea have been identified as an area with the greater potential, and Deep Sea Ocean Mission on studying the same has been initiated by MIS. Specific programs are also available with BRAC for promotion or promotion of translating technologies at different levels. Important point in this line is the bridging the gap between the industries and academia to capitalize and monetize the science and technology fraternity and address the market requirements and achieve the sustainable development goals. It is important to interact between different agencies to develop a combined approach for developing the ecosystem which support achieving the targets in time. It is proposed that a wish list of research priorities which may be gathered from the industries in marine biotechnology, wherein they think that the collaboration and the academic involvement is required and is important. CSIR NIO, we know, is working in the area since in this marine biotechnology. Several programs have been operated in the past and have access to oceans and facilities for leading the marine biotechnology industry, academia, interaction program. So in the first meeting and panel, the talks and panel discussions, it came forward that CSIR NIO may take a lead role in organizing a brainstorming session on identifying the areas where industry institution collaborations in marine biotechnology are required and identify the potential scientists and industries or institutions from India to be included in these projects. And this proposal or this may be proposed for inclusion in the National Marine Mission, which is under the consideration of Government of India. We trust, I think we covered all the points, and we trust that that the engagement that you had shown today will be continued in the months ahead, so that we can take this the outcomes of this iconic meeting to a successful story. So thank you very much.
Thank you, Dr. Anas, for summing up these fruitful discussions. I now call upon Dr. Rahul Kaushik, scientist, CSIR NIO, to give the vote of thanks.
Thank you, ma'am, and good afternoon to all. It gives me immense pleasure to propose a vote of thanks on this occasion. First of all, we would like to thank our keynote speaker, Professor Tapas Kumar Kundu, Professor JNCASR Bengaluru, for his informative lecture on marine tunicates, cyanobacteria, fungi, seaweeds, and FDA-approved drugs for therapeutic applications. We are also grateful to our plenary speakers, Dr. R. Dinesh Kumar, Senior Scientist, CSIR, CSM, CRI, Dr. Rahul Wake, Director, High Media Laboratories Private Limited, Dr. Shantanu Das Gupta, Senior Vice President, Head of Synthetic Biology and Algae to Oil R&D, Reliance Industries Limited, Dr. Sridhar Narayan, Director, Foundation for Neglected Disease Research, and Dr. Shesh Raghukumar, Managing Director, Mycotech Private Limited, for providing so much value to our audience. We extend a very hearty note to a vote of thanks to all the panel members, Dr. Vijay Kumar, Scientist G, Ministry of Earth Sciences, Dr. Dheeraj Kumar, Senior Manager, Technical, DBT Birec, Dr. Girish Mahajan, Senior Vice President, High Media Laboratories Private Limited, Dr. VV Sanil Kumar, Chief Scientist and Head, Business Development Group, CSIR NIO, Commander Ramneet Kaushik, Scientist, FDST Technology Development Board, Dr. Taslim Arif Sayed, CEO and Director, CCAMP, Shri Tapan Chattopadhyay, ex-Deputy Director, Indian Chamber of Commerce, Shri Hemant Kulkarni, Senior Principal Scientist, CSIR TMD, and Dr. Belle Damodar Shinoy, Principal Scientist, CSIR NIO, for sharing with us your opinion on way forward with marine natural products and to strengthen the collaboration between the CSIR NIO and the research and development industries. We would like to express our sincere thanks to DG CSIR, Dr. Rajesh Gokhale, Professor VM Tiwari, E3OW Theme Director and Director, CSIR NGRI Hyderabad, Dr. Vibha Malhotra Shawnee, Head, CSIR IP Unit, and Dr. Devendra Singh, Principal Scientist, CSIR TMD, for their support in planning this event. We also thank our Director, CSIR and IO, Professor Sunil Kumar Singh, for his continuous suggestions and guidance to shape this event. We are also grateful to our ITG head, Dr. Yatish, Principal Scientist, and his technical team members, Mr. Akshay, Mr. Ryan, Mr. Jay Singh, the Director Office, and NFDC team in New Delhi for their technical support. We would also like to acknowledge Mr. Venkat Krishnamurthy and all the BioProSmart project members for their valuable contributions. Finally, a big thank you to all the participants who are attending this event. Have a great day. Thank you.
Thank you, Dr. Kaushik. We have a small announcement. Tomorrow, we have a hybrid iConnect event on aquaculture from 10 a.m. to 1:30. And those interested in attending it are welcome to join this event. Once again, a big thank you to all of you. Stay safe and have a pleasant afternoon. Jai Hind.